For most of human history, rock was just rock. It was something to break, carve, stack, and build with. Stone raised walls, paved roads, lined canals, and revealed veins of metal hidden inside mountains. People worked with it long before they ever tried to understand it, and yet, beneath that practical familiarity, the earth had already been writing its autobiography in silence. Every layer of sediment, every fossil embedded in limestone, every dark vein of igneous rock cutting through older strata was part of a record far older than language. The problem was not that the record was missing. The problem was that no one yet knew how to read it. A great change began in the 18th and 19th centuries when natural philosophers slowly became geologists and the earth itself became an object of disciplined study. This was more than a scientific shift. It was a profound change in how people imagine time. The old world had largely explained natural history through inherited doctrines, often shaped by theology, classical tradition, and a belief that the major features of the earth had been formed in a small number of dramatic moments. But as field observations accumulated, as mining expanded, as roads and canals cut deeper into the land, and as fossils were collected in increasing numbers, the ground beneath those older explanations began to move. Early geology did not begin in lecture halls, stream beds, sea cliffs, and construction sites. People noticed that rocks were not arranged randomly, they were layered. Some layers contained shells, others contained plant impressions. Some were coarse and full of pebbles, while others were fine, compacted, and smooth. This suggests order. It suggested sequence. It suggested that the earth had a history. One of the earliest major attempts to explain that history came through a theory known as Neptunism. Developed in the late 18th century by Abraham Werner and influenced by earlier ideas from Johann Gottlob Lehmann, Neptunism proposed that most rocks had formed from a primordial ocean that once covered the Earth. According to this view, the planet had originally been surrounded by a universal fluid, and as this vast body of water gradually receded, different rock types precipitated out in sequence. Mountains, plains, and even many hard crystalline rocks were interpreted as deposits left behind by that ancient sea. It was an elegant theory, and for a time, a persuasive one. It explained layered rocks in a way that felt orderly and grand. It also fit comfortably with flood traditions familiar to many Europeans, making it intellectually attractive in an age when science and theology still overlapped heavily. But Neptunism had limits. It could describe rock layers, yet struggled to explain volcanoes convincingly. Igneous rocks posed a particular problem. If all major rocks had formed through precipitation from water, then what should one make of molten material erupting from deep within the earth? The theory had to treat volcanic activity as secondary, local, and relatively unimportant. That interpretation would not survive for long. A different vision emerged through the work of James Hutton, a Scottish thinker whose observations transformed geology. Hutton did something simple and revolutionary. he trusted the evidence of ordinary processes. As he moved through the Scottish landscape, he studied erosion along seams, sediment carried by rivers, and the layered rocks exposed in cliffs and plains. He noticed that the same natural forces operating in the present, weathering, transport, deposition, uplift, could explain the formation of the Earth's surface if given enough time. That idea changed everything. It meant that the world did not need to be explained through a few singular catastrophes or a one-time global flood. It could be understood as the result of continuous natural processes working over immense spans of time. This became the heart of uniformitarianism. The phrase most closely associated with Hutton's view is often summarized as the idea that the present is the key to the past. That did not mean the Earth never changed dramatically. It meant that the forces shaping it were not mysterious or entirely different from those we can still observe now. Mountains rise, then erode. Sediments accumulate, harden into rock, and later may be lifted again. The planet is not static, but neither is it chaotic. It is dynamic through continuity. Hutton's theory also carried a far more unsettling implication. The Earth had to be vastly older than many people had assumed. If landscapes were shaped by slow erosion and gradual deposition, then the time required was immense. Hutton famously suggested no trace of a beginning and no prospect of an end. In a world still deeply influenced by short biblical chronologies, that was a radical statement. He was not merely offering a new geological theory, he was expanding time itself. While this debate unfolded, fossils introduced another challenge. For centuries, strange stone shapes resembling shells, fish, and bones had been found in rock layers, sometimes high in mountains or far from the sea. Some dismissed them as curiosities, tricks of nature, or even deliberate deceptions placed in stone. But such explanations grew harder to defend as collections became more systematic. Fossils were not random shapes. They showed structure, repetition, and pattern. They resembled living organisms too closely to be ignored. Georges Cuvier played a crucial role in changing that understanding. By studying fossils carefully, especially vertebrate remains from the Paris Basin, he concluded that many creatures preserved in rock no longer existed anywhere on Earth. This was a profound realization. Extinction was real. Species were not fixed inhabitants of a stable world stretching unchanged from creation to the present. They could disappear. Entire faunas could vanish and be replaced by others. Cuvier did not accept evolution in the modern sense, but his work strongly supported catastrophism, the idea that Earth's history included episodes of sudden destruction. He argued that some fossil assemblages reflected worlds that had been wiped out by major events, after which new species appeared. This view challenged simple continuity, yet it also helped establish a key truth. The Earth's biological history was not one unbroken line, it was interrupted, broken, replaced. So geology advanced through tension. Neptunus emphasized ancient seas. Huttonians emphasized gradual processes. Catastrophists emphasized sudden destruction visible in the fossil record. Over time, the field matured not by choosing myth over evidence or theory over observation, but by forcing every explanation to answer to the rocks themselves. That transformation accelerated in the 19th century. Charles Lyell became one of the great organizers and defenders of uniformitarian thinking, presenting it with clarity and discipline in ways that influenced generations of geologists. At the same time, William Smith made another breakthrough by recognizing that rock layers could be identified and correlated by their fossils. Strata in different regions might look similar, but fossils offered something more precise. Certain layers consistently contained characteristic fossil assemblages, and these assemblages could be used to match rocks of the same relative age across wide distances. This became the principle of fossil succession. That principle helped turn scattered local observations into a global framework. Geologists could now compare strata from different regions, arrange them in relative order, and build something larger than a map of isolated outcrops. They could begin assembling a time stratigraphic column, a sequence of Earth history written in stone. Layer by layer, fossil by fossil, the planet's chronology came into focus. And with that, the Earth stopped being a static backdrop. It became a story. Not a story told by memory, legend, or scripture alone, but by sediment, erosion, burial, uplift, and extinction. The planet had a past that could be reconstructed. Its surface was not merely scenery. It was evidence. Its rocks were not mute. They were archives. Humanity had finally begun to realize that beneath every cliff and beneath every valley lay not just material, but time itself, compressed, fractured, preserved, and waiting for someone patient enough to read it. Once geologists accepted that rocks preserved a sequence, the next challenge was far more difficult. How do you put that sequence in order, and how do you compare one place on Earth with another? A cliff face might reveal a beautiful stack of layers, but a single outcrop could only tell a local story. To transform geology into a history of the whole planet, scientists needed principles that could turn isolated exposures into a coherent timeline. That effort produced one of the most elegant intellectual frameworks in science, a way of reading relative time directly from stone. One of the first and most powerful ideas was the principle of superposition. In a sequence of undisturbed sedimentary rocks, the oldest layer lies at the bottom and the youngest at the top. The logic seems almost obvious now, but at the time, it was transformative. Sediments settle layer upon layer. If those layers remain largely intact, their vertical order preserves the order of their formation. A cliff is no longer just a wall of stone, it becomes a timeline. The bottom is deeper in the past, the top is closer to the present. With that single principle, geologists gained a way to reconstruct the order of events in a rock sequence, even when they had no idea how many years separated one layer from the next. Another key insight came from the original horizontality. Sediments are typically laid down in roughly horizontal layers, so when geologists encounter rock beds tilted, folded, or standing nearly vertical, they know those rocks were changed after their formation. The distortion itself becomes evidence of later tectonic events. A flat seabed becomes buried, hardened into rock, uplifted, compressed, bent, and exposed again. Suddenly, a deformed rock layer is no longer just a shape, it is an event sequence frozen in structure. First deposition, then lithification, then deformation, then uplift, then erosion. Geology began to reveal not only what existed, but what happened. From there came the principle of cross-cutting relationships. If one rock body cuts through another, the cutting feature must be younger. An igneous intrusion slicing across sedimentary layers, a fault offsetting an older formation, a volcanic vein running through pre-existing rock. Each one tells the same story. Something had to exist before it could be cut. This idea gave geologists a way to untangle more complex histories where different processes overlapped and disturbed the neat simplicity of layered sediment. It also dealt a serious blow to theories that imagined the Earth as largely static after its initial formation. Cross-cutting relationships made it impossible to ignore repeated episodes of intrusion, movement, breakage, and renewal. Yet, even with these principles, one major problem remained. You could determine which rock was older and which was younger in a single area, but how could you compare rocks across distant regions? A sandstone in one country might look like a sandstone in another, yet appearance alone could be misleading. Texture and color were not enough. What geology needed was a more reliable marker, something embedded in time itself. That marker was found in fossils. William Smith, working not as an abstract theorist, but as a practical surveyor and engineer, noticed that particular layers of rock consistently contained particular kinds of fossils. More importantly, those fossil assemblages appeared in a predictable order. Even when the rock itself changed in appearance from one region to another, the fossil sequence remained meaningful. This became the principle of fossil succession. Strata can be identified and correlated by their fossil content because biological communities changed throughout time in recognizable ways. This was a stunning breakthrough. It meant geologists could match layers across wide geographic distances even when the rocks were not identical in composition. Fossils became clocks of relative time, not because they gave a numerical age, but because they revealed position in the grand sequence of life. With superposition and fossil succession working together, geologists could begin assembling a global history from local pieces. One outcrop might preserve marine shells, another trilobites, another early vertebrates, and another mammalian fossils, each embedded in its own set of layers. Correlate enough of these sections, and a broader time stratigraphic column begins to emerge. This effort eventually gave rise to the geologic timescale, one of the greatest organizing structures in science. The immense history of Earth was divided into major units, eras such as the Paleozoic, Mesozoic, and the Cenozoic. then further into periods and epochs. These divisions were not arbitrary. They reflected real changes in the fossil record and in the character of the rocks themselves. Boundaries in the scale often marked moments of profound biological turnover, environmental transition, or extinction. Time was no longer just an abstract abyss, it had architecture. And still, a deeper question remained unanswered. Relative age is powerful, but it is not the same as absolute age. Knowing that one layer is older than another does not tell you whether the gap between them is 1,000 years, 1,000,000, or 100,000,000. For a long time, this was the central unresolved problem in geology. The principles of stratigraphy had revealed sequence, but not duration. Geologists could say which events came first and which followed, but not how far back the beginning truly lay. In the 19th century, many attempts were made to solve that problem, but most were flawed. Some estimates relied on rates of cooling for the Earth, others on sediment accumulation, and others on ocean chemistry. These methods seemed promising, but they depended on assumptions that were far too simple for a planet as complex as ours. The results varied wildly. The Earth might be millions of years old, perhaps tens of millions, perhaps more, but no method could give a secure answer. The rocks had revealed vast age, yet the number itself remained elusive. The breakthrough came in the early 20th century with the discovery that radioactive decay could serve as a natural clock. This was revolutionary because it introduced a process rooted not in surface change, which can vary greatly, but in the internal properties of atoms themselves. Certain elements are unstable. Over time, they decay into other elements at a fixed statistical rate. That rate is expressed through half-life, the time required for half of a given quantity of the radioactive parent element to transform into its daughter product. Unlike erosion, climate, or sedimentation, this process is not easily altered by ordinary chemical or physical conditions. It offers something geology had never possessed before, a clock built into matter. Radiometric dating transformed Earth history from a relative narrative into a measurable one. By examining minerals that formed when a rock crystallized and by measuring the ratio between parent and daughter isotopes, scientists could calculate how much time had passed since that mineral formed. Uranium decaying to lead, potassium to argon, and other isotopic systems opened a new window onto deep time. Suddenly, the vague immensity proposed by Hutton and defended by Lyell could be quantified. The Earth was not merely very old, it was about 4.6 billion years old. That number changed everything. It confirmed that the timescales required by geology and evolution were not exaggerations, but necessities. Mountains could rise and erode, oceans could open and close, species could emerge, diversify, and vanish. Entire faunas could dominate for tens of millions of years and then disappear into rock. The age of the Earth was no longer a matter of philosophical speculation. It had become a measurable property of the planet itself. And with that, geology completed one of the most profound revolutions in human thought. The Earth was no longer a young stage with a short, simple past. It was ancient beyond ordinary imagination, shaped by slow processes, interrupted by violence, and recorded in layers that preserved both continuity and catastrophe. Rock was no longer just material, it was memory. Fossils were no longer curiosities, they were witnesses. Time itself was no longer hidden behind myth or assumption, it had been brought into view through observation, inference, and the patient reading of stone. What makes this story so powerful is not just that humanity learned the earth was old, it is that humanity learned how to know it. The cliffs, the fossils, the intrusions, the tilted strata, the radioactive minerals, none of them spoke in words, yet together they revealed a history more vast than any civilization, more dramatic than any legend, and more humbling than any myth. The surface beneath our feet is not fixed. It is the surviving edge of an unfinished story, one written over billions of years and still being added to now. By the time geology matured into a modern science, the Earth had changed in human imagination forever. It was no longer merely the place where history happened, it had a history of its own, and once that truth was seen, it became impossible to look at a mountain, a canyon, or a fossil-bearing cliff in quite the same way again. There are ideas so powerful that, once understood, they seem almost inevitable. Evolution is one of them. Today, it feels woven into the way we see life itself, as if the world had always been waiting for someone to say it clearly. But when Charles Darwin first stepped aboard the HMS Beagle in 1831, evolution was not yet the obvious answer. It was a dangerous possibility, half-formed, surrounded by older beliefs about fixed species and special creation. Darwin did not leave England as a revolutionary. He left as a curious young naturalist trained to observe, collect, compare, and record. What changed him was not a single revelation, but a slow accumulation of evidence gathered across oceans, islands, fossils, climates, and living forms. That is what makes Darwin's story so compelling. It is not the story of a man inventing nature from imagination. It is the story of a man being cornered by reality. Born in 1809 into a prosperous English family, Darwin was expected to follow a respectable path. Medicine did not suit him. The clergy did not truly claim him either. Yet he possessed something far more important for what was coming: patience, discipline, and an unusually alert eye for pattern. By the time he joined the Beagle, he had already absorbed much of the scientific spirit of his age. He knew how to look carefully. He knew how to compare details. What he had not yet done was fully question the assumption that species were permanent. The voyage changed that. As the Beagle moved around the world, Darwin encountered a pattern that was too consistent to ignore. Species were not distributed randomly. The plants and animals of one region resembled one another in ways that made geographic sense, even when similar environments existed elsewhere. A desert in one part of the world did not contain the same creatures as a desert in another. The tropical regions of South America did not resemble the biota of Africa or Australia, even when climate seemed comparable. This raised a troubling question. If environments alone shaped organisms, Why were similar habitats occupied by different kinds of life in different regions? Why did geography seem to matter as much as climate, perhaps more? Darwin began to see that barriers such as oceans, mountains, and distance were not just obstacles to movement, they were engines of divergence. Populations isolated for long periods could slowly become different from one another. This was not yet the full theory of evolution, but it was a crack in the old idea of fixed creation. Species no longer looked like perfectly independent units placed arbitrarily across the world. They looked connected to place, history, and separation. South America deepened that suspicion. Darwin was struck not only by the living animals he saw there, but by the fossils emerging from the ground. This was one of the most important moments in the formation of his thought. In the fossil beds of South America, extinct mammals appeared that resembled, in broad form, animals still living on the same continent. Fossil armor-plated creatures suggested a relationship to living armadillos. Other extinct forms echoed the shape of creatures like sloths and camel-like mammals found in South America. The resemblance was not exact, but it was close enough to be unsettling. If species had been separately created in fixed forms, why should the dead inhabitants of a continent resemble its modern living ones more than creatures found elsewhere? Why should the past and present of a single place seem linked? This was one of Darwin's greatest insights. The fossil record was not simply a graveyard of vanished organisms. It looked like a sequence. Extinct forms seemed related to later living forms in the same region. The Earth's history was beginning to look less like a series of disconnected acts of creation and more like a branching continuity through time. Then came the Galapagos. Few places in the history of science have become as symbolic as these volcanic islands, and for good reason. Lying hundreds of miles off the coast of South America, the Galapagos offered Darwin a natural laboratory of isolation. These were young volcanic islands born of fire, never connected to the mainland, and yet inhabited by plants and animals clearly related to South American life. That alone was suggestive. It implied dispersal, colonization, and survival across barriers that seemed formidable. But the deeper revelation came from variation within the islands themselves. The giant tortoises differed from island to island. Some had shell shapes and neck proportions that seemed suited to different conditions. On one island, a longer neck might help an animal reach sparse vegetation higher above ground. On another, a different form seemed sufficient. The tortoises were close relatives, unmistakably linked, yet they were not identical. Geographic isolation had not merely preserved them, it had shaped them. The same was true, even more famously, among the finches. Darwin did not immediately grasp the full significance of these birds during the voyage, but the pattern would later become central to his thinking. The finches of the Galapagos shared a common overall structure, yet their beaks varied in remarkable ways. Some were thick and strong, suited to cracking seeds. Others were narrower and finer, better for insects or cactus feeding. These were not random differences. They were functional. They suggested that one ancestral stock, once isolated across different islands and ecological opportunities, had diversified into multiple forms, each adjusted to a particular way of life. That idea would become one of the most elegant illustrations of adaptive radiation, a single lineage entering new environments with little competition, then spreading into available niches and gradually diverging as small advantages accumulate over time. What made this so powerful was not simply that species differed. Naturalists already knew that variation existed. The deeper point was that variation seemed directional when placed in a particular environmental and geographic context. On islands with limited space and resources, even slight differences could matter. A somewhat stronger beak, a slightly different feeding habit, a small shift in behavior. Those differences could influence survival and reproduction. Given enough time, such changes would not remain small. Darwin was beginning to understand that life might not be fixed at all. It might be plastic, responsive, historical. And if that was true, then the living world was far more dynamic than anyone had imagined. Before Darwin, many people could accept extinction as Cuvier had shown, or accept deep time as Lyell had argued, yet still resist evolution itself. Species could vanish, the earth could be old, but organisms themselves were still often imagined as fundamentally fixed. Darwin's genius was not simply in seeing one new fact, it was in bringing several lines of evidence into contact with one another. Geography suggested separation and divergence. Fossils suggested continuity between past and present forms. Island life suggested that isolation could sharpen differences rather than preserve sameness. Together, these observations began to point in one direction. Life changes, not suddenly, not theatrically, but gradually through accumulation. By the time Darwin returned to England, the foundations of his old worldview had already been weakened. He did not yet have every mechanism worked out. He would spend decades refining the idea, testing it, comparing evidence, and struggling with objections. But the crucial shift had already happened. The natural world no longer looked like a collection of fixed creations placed neatly across the globe. It looked historical, related, mutable, alive not only in the present, but in time. The voyage of the Beagle did not give Darwin a finished theory. It gave him a problem he could never again unsee. And once that happened, the living world was no longer static scenery. It had become a moving story. Seeing that species were not fixed was only the beginning. The far harder question was mechanism. It was one thing to suspect that living forms had changed over time, branching from earlier ancestors and adapting to different environments. It was another to explain how such change could happen without direction, without a designer stepping in for every small adjustment, and without the world descending into biological chaos. Darwin returned from the Beagle with evidence powerful enough to unsettle old beliefs, but not yet enough to complete a theory. For years, he carried the problem with him, turning it over from every angle, comparing fossils, living species, geographic patterns, and the structure of variation itself. He was no longer asking whether life had changed. He was asking what force could slowly sculpt that change across immense spans of time. The Galapagos finches would later become one of the clearest illustrations of that answer. These birds were not dramatic creatures. They were small, easily overlooked, variations on a familiar theme. Yet in their beaks lay one of the most elegant clues in all of biology. On different islands, finches with closely related bodies showed different feeding structures. Some had thick, crushing beaks suited for seeds. Others had finer beaks, better for insects. Still others were adapted for cactus feeding or other available resources. The important point was not simply that they were different, it was that the differences made ecological sense. Form matched function, structure reflected opportunity. When a few ancestral birds arrived in a place with open niches and limited competition, their descendants did not remain identical. Over time, small variations that gave an advantage in one environment became more common there, while different variations succeeded elsewhere. What began as one lineage could diverge into several. This was not random disorder, it was organized divergence shaped by survival. Darwin came to understand this as a broader pattern, later called adaptive radiation. A species enters a new region where there is little competition and many unused ways of living. Within that setting, populations begin to vary. Some remain in the original niche, others spread outward into different ones. Slight changes accumulate generation after generation until the descendants become distinct forms adapted to different diets, different habits, different pressures. The Galapagos were powerful not because they created evolution, but because they made its logic visible in compressed form. Still, even this did not fully solve Darwin's problem. Variation existed. Isolation mattered. Geography shaped life. But why should certain variations persist while others disappeared? Why did adaptation move in a direction that seemed fitted to local conditions? The breakthrough came when Darwin encountered the idea of Thomas Robert Malthus. Malthus had argued that human populations tend to increase faster than the resources available to support them, and that this creates inevitable struggle through famine, disease, war, and competition. Darwin immediately recognized that the principle applied far beyond humans. In fact, it applied even more strongly to the rest of life. Most organisms produce far more offspring than can possibly survive. A fish may release thousands of eggs. Plants scatter vast numbers of seeds. Insects multiply rapidly. Even animals with relatively few young still produce more individuals than the environment can support in the long run. If all survived and reproduced, the Earth would be overwhelmed. Yet this does not happen. Most die. Most never reproduce. That simple fact transformed Darwin's thinking. If more individuals are born than can survive, then life is necessarily shaped by a constant, mostly silent struggle. Not always open combat, not always predator against prey, but competition for food, space, shelter, mates, and survival itself. Within that struggle, any inherited variation that gives even a slight advantage can matter. A bird with a beak slightly better suited to local food may survive a hard season when others do not. A plant slightly more tolerant of drought may leave more seeds. A mammal whose coloring gives better concealment may escape long enough to reproduce. The advantage need not be dramatic. Over many generations, tiny differences are enough. This became natural selection. Darwin did not imagine it as a conscious force. It has no purpose, no foresight, no end goal. It is simply the outcome of variation, inheritance, and unequal survival. Individuals vary. Some of those variations affect survival and reproduction. Those that reproduce pass on more of their traits. Over time, populations change. If the process continues long enough, small changes can become large ones. Varieties become distinct forms become species. The tree of life branches outward. What made this idea so powerful was that it linked countless observations into one coherent structure. The fossil record showed continuity between extinct and living organisms. Geographic distribution showed that closely related forms tend to occur near one another rather than in whatever environment happens to suit them best. Islands revealed how isolation sharpens difference. Artificial selection practiced by breeders of pigeons, dogs, sheep, and crops showed that dramatic change could be produced by selecting small variations generation after generation. Darwin saw that nature, without intention, could do something similar on a far larger scale and over vastly greater periods of time. He called this descent with modification. That phrase matters because it captures both continuity and change. Species do not appear fully formed and isolated from one another. They descend from earlier forms and gradually become modified through time. The result is not a ladder with life climbing toward a fixed summit, but a branching pattern. Some lineages diversify. Some remain relatively stable for long periods. Some disappear. Extinction is not an exception to evolution. It is part of it. The history of life becomes a tree, not a chain. This was one of Darwin's deepest breaks from older thinking. Traditional ideas often treated species as separate creations, each with a fixed essence. Darwin replaced that vision with ancestry, divergence, and branching relationship. Modern horses could be seen as modified descendants of earlier forms, not independent creations. Mammals, birds, reptiles, fishes, and countless invertebrates could all be placed within an immense unfolding history. Life became historical in the strongest possible sense. To understand any organism, one had to ask not only what it is, but what it came from. Darwin knew his theory was not complete. He recognized serious problems. He did not understand heredity in a modern genetic sense. He could not fully explain how traits were transmitted or how new variation arose. He knew the fossil record was incomplete. He understood that gradual evolution required immense time and that many critics would resist such a scale. He was aware, too, that natural selection might be misunderstood as crude or cruel, when in fact it was simply the unavoidable consequence of overproduction and differential survival. That is part of what makes Darwin such a compelling figure. He was not casually confident. He was careful, even burdened by the magnitude of what he was proposing. He spent more than 20 years refining his arguments before publishing On the Origin of Species in 1859. When it appeared, the book did not merely offer a biological theory, it changed the intellectual architecture of life itself. It challenged fixity, redefined humanity's place in nature, and transformed biology from a science of static forms into a science of historical change. Its influence spread because it explained so much at once. Why do island species resemble nearby mainland forms? Why do fossil species resemble later living ones in the same region? Why do similar environments contain different organisms on different continents? Why do structures seem fitted to ways of life without requiring each one to be separately designed from scratch? Darwin's theory did not answer every question, but it gave these scattered facts a common logic. And once that logic was seen, it became very difficult to return to the older view. The world no longer looked like a museum of fixed types. It looked like a living experiment unfolding across deep time. Every species carried history within it. Every feather, fin, claw, tooth, and leaf became evidence not just of function, but of inheritance. Similarities between organisms were no longer merely convenient categories, they were clues to relationship. In that sense, Darwin did more than explain evolution. He changed how nature could be seen. Life was no longer a collection of isolated forms. It was a dynamic web of descent, adaptation, struggle, and divergence. The grandeur of that vision lies not in drama, but in accumulation, small differences, long spans of time, endless branching. And from that slow, patient process came the overwhelming diversity of the living earth. The voyage of the Beagle had opened the question. Natural selection gave it force. Descent with modification gave it structure. And from those ideas emerged one of the most powerful realizations in science. Life does not simply occupy the world. It becomes it, generation by generation, shape by shape, branch by branch. There is something deeply counterintuitive at the heart of evolution. We tend to imagine change in nature as the story of a single animal struggling, surviving, and passing on its strength, as if evolution were written through individual triumph. But population genetics shifts the entire frame. It tells us that the true unit of evolutionary change is not the lone organism, but the population. An individual is born, lives, reproduces, or fails to reproduce, and dies. A population endures. A species persists or disappears not because one creature succeeds, but because the genetic composition of the group changes across generations. That idea is where modern evolutionary thinking becomes both more mathematical and more fascinating. Mendelian genetics had already shown that traits are inherited through discrete units, what we now call genes. But population genetics asked a much larger question. What happens when those genes are spread across thousands or millions of individuals, all breeding, moving, surviving, and dying under different conditions? Once that question is asked, life stops looking like a collection of separate bodies and starts looking like a dynamic pool of inherited possibilities. A population, in the simplest sense, is a group of organisms of the same species living and potentially breeding within a given area. It could be small and local, or vast and nearly continuous. What matters is not the exact size, but the fact that the members share a gene pool. Every gene present in every member of that population contributes to that pool. This is one of the most elegant ideas in biology because it turns inheritance into something collective. Traits do not just belong to individuals, they exist as frequencies within populations, rising and falling over time. That brings us to one of the key concepts in population genetics: gene frequency, or more precisely, allele frequency. If a gene exists in different forms, those forms are called alleles. In a population, one allele might be common, another rare. Population genetics asks a deceptively simple question: how common exactly? What fraction of all copies of a gene in the population belongs to one allele, and what fraction belongs to another? This is not merely bookkeeping. Once the numerical incidence of alleles is known, the hidden structure of evolutionary change begins to appear. Imagine a gene with two alleles, A and A. If A has a frequency of 0.99, then A must have a frequency of 0.01. Together, they add up to the whole. This sounds almost trivial, but it gives biologists a way to describe inheritance not as a family story, but as a population-level pattern. The same is true for genotype frequencies. If the frequency of one allele is known, the distribution of combinations AA, AA, and AA can be predicted under certain conditions. Suddenly, the invisible architecture of heredity becomes measurable. This is where Hardy-Weinberg equilibrium enters the story, and few ideas in biology are more quietly powerful than this one. Hardy and Weinberg showed that an idealized population, where mating is random and no evolutionary forces are acting, allele frequencies remain stable generation after generation. The population becomes, in effect, a still surface, no drift, no selection, no mutation changing the balance, no migration altering the pool. In that equilibrium, genotype frequencies follow a predictable pattern: p squared, 2pq, and q squared. On paper, it looks almost too neat. In nature, it almost never exists perfectly. And that is exactly why it matters. Hardy-Weinberg is important not because the real world obeys it completely, but because it provides a baseline. It tells us what a population would look like if nothing were changing. Once real populations depart from that condition, the deviation itself becomes evidence. Something is acting. Some force is pushing gene frequencies away from stillness. The equation does not just describe stability, it reveals change by showing what stability would have looked like. One of the most interesting consequences of this framework is that populations can carry far more hidden variation than their visible traits suggest. A recessive allele may be rare in the phenotype, but far more common in the gene pool, sheltered inside heterozygous individuals. In a population, the outward appearance of rarity does not always mean genetic insignificance. This creates a kind of hidden reservoir of variation, a storehouse of possibilities that may remain invisible for generations until environmental change, random chance, or altered breeding patterns bring it forward. Evolution, in this sense, is not just the visible shift in bodies, it is also the silent rearrangement of genetic potential. The classic example of coat color in rodents captures this beautifully. A population living on dark rock may come to be dominated by dark-coated individuals, while the same species on pale sand remains largely light-colored. At first glance, this looks like a simple visual contrast, but population genetics reveals the deeper mechanism. If predation is easier against animals that stand out from their background, then selection gradually changes the frequency of the alleles that control coat color. Dark variants survive more often on dark substrate. Light variants survive more often on pale substrate. Over time, the gene pool itself shifts. What changes is not merely the appearance of a few animals, but the numerical structure of heredity across the whole breeding population. That distinction matters because it turns natural selection into something far more precise. Selection does not reach into nature and choose individuals in a conscious sense. It alters reproductive success, and through that, it alters gene frequencies. The result may look like adaptation in fur color, body form, or behavior, but beneath every visible pattern lies a statistical shift in inheritance. Population genetics also changes how we think about success and failure in evolution. The death of one organism may feel dramatic, but from the viewpoint of the gene pool, it matters only insofar as it affects transmission. A trait is not favored because it is beautiful, noble, or impressive. It is favored because it survives the arithmetic of reproduction. If an organism fails to leave offspring, its genes leave with it. If another leaves many, its alleles become more common, even if the difference began as something slight. Evolution becomes less like a heroic narrative and more like a quiet sorting process repeated millions of times. This is what makes population genetics so compelling. It strips away the illusion that nature changes through grand gestures alone. Instead, it reveals that evolution often works by tiny statistical imbalances accumulating over time. A slight difference in survival, a minor shift in mating, a small reproductive advantage. Generation after generation, these imbalances reshape populations until the change becomes visible in flesh, form, and function. What emerges is a completely different image of life. A species is no longer just a collection of similar-looking individuals. It is a moving genetic structure, a living distribution of possibilities, always being tested, filtered, and rearranged. Beneath every forest, desert, reef, and tundra lies this invisible game of numbers, frequencies, and inheritance. Natural selection may be the force most people remember, but population genetics shows where that force actually leaves its mark, not only in bodies, but in the changing proportions of genes carried by the population as a whole. And that may be the most important shift of all. Evolution stops being a story about isolated creatures and becomes a story about pools of variation, about continuity across generations, about populations balancing on the edge between stability and change. The individual matters, of course, but only as part of something larger. In the end, Nature does not remember one life for very long. It remembers what that life adds to the pool. If Hardy-Weinberg equilibrium gives us the image of a population at rest, then the real drama of evolution begins when that rest is broken. In nature, stability is almost never perfect. Populations do not live in sealed mathematical worlds. They live in shifting landscapes, under changing climates, among predators, barriers, accidents, migrations, and mutations. The gene pool is constantly exposed to forces that can alter its structure, and once allele frequencies begin to move, evolution is no longer an abstract concept. It becomes measurable. One of the most familiar of these forces is natural selection. In population genetics, natural selection is not just the survival of a well-adapted individual, it is the steady increase of alleles that improve reproductive success and the decline of alleles that reduce it. This is why selection is so powerful. It does not merely remove organisms, it edits the gene pool. A population living on dark volcanic rock may gradually come to carry more of the alleles for dark coloration because individuals that blend into the background are less likely to be taken by predators. On pale sand, the opposite may happen. Over time, the visible trait changes because the underlying frequencies change first. Selection, then, is not simply visible in bodies, it is written into the arithmetic of inheritance. Mutation plays a very different role. Selection sorts what already exists, but mutation introduces novelty. Most mutations are small, many are neutral, and a great number are harmful when expressed. Some are recessive and remain hidden in heterozygous carriers, shielded from immediate elimination. Others are lethal enough to remove themselves from the population almost at once. This creates what geneticists call genetic load, the burden of deleterious alleles carried within a population, often invisibly. At first glance, this may sound like a flaw in life's design, but it is also part of how evolution remains open-ended. Mutation is the source of raw material. Without it, there would be nothing new for selection to favor, reject, or preserve. Life would reach equilibrium and stay there, stable perhaps, but trapped. The fascinating tension is that mutation and selection often work in opposite directions. Mutation continuously introduces new variation, including harmful alleles, while selection works to remove what lowers survival or fertility. In some cases, the two forces can reach a kind of balance. Harmful recessive allele may continue to exist in a population because mutation keeps reintroducing it, while selection removes only those individuals in whom it is fully expressed. The result is not perfection, but a dynamic compromise. Populations are never genetically pure. They are always carrying hidden variation, some useful, some costly, some waiting for circumstances to decide their fate. Gene flow adds another layer to this story. Populations are often treated as if they are separate, but in reality, organisms move, individuals migrate, pollen travels, seeds disperse, animals cross boundaries that maps would call distinct. When genes move from one population into another, allele frequencies shift. This can reduce differences between groups, blending what isolation had begun to separate. Gene flow acts like a kind of biological mixing current, preventing divergence from going too far, too fast. Yet when gene flow is reduced or stopped, populations begin to drift apart more easily. At that point, geography becomes more than scenery, it becomes destiny. Geographic isolation is one of the most important pathways by which populations begin to split into something new. A river changes course and divides a population. A valley deepens. A mountain rises. A climate shift expands a once small barrier into an effective wall. Suddenly, two groups of the same species are no longer exchanging genes. They still began with nearly the same gene pool, but from that moment forward, they experience mutation, selection, and random fluctuations separately. Their environments may differ slightly. Their predators may not be identical. Their breeding seasons may shift. Their small differences accumulate. At first, the divergence is minor, then noticeable, and eventually profound. What began as one population can, over time, become two. This process does not always require dramatic barriers. Sometimes spatial isolation emerges across a broad geographic range where members of a species are distributed continuously, yet actual interbreeding becomes rare over distance. Local environments begin to shape local gene pools. Insects with short flight ranges, for example, may remain effectively isolated even across relatively modest environmental discontinuities. Over time, geographic races can form, populations still belonging to the same broader species, yet genetically distinct enough that the boundary between race and new species begins to blur. This is one of the most intriguing moments in evolution, when sameness begins to fracture but has not yet fully broken. Then come isolating mechanisms, the final barriers that transform divergence into speciation. These mechanisms can prevent interbreeding before fertilization even occurs. One population may breed in a different season from another. Courtship signals may no longer match. Habitat preferences may separate individuals so effectively that contact becomes rare. Even when mating occurs, hybrid inviability or infertility may block the formation of a stable shared lineage. The mule remains one of the clearest examples in biology. A horse and a donkey can produce offspring, but that offspring is generally sterile. Gene exchange stops there. The hybrid exists, but does not reconnect the gene pools. Reproductive isolation is what finally seals the split. What makes this so compelling is that species formation is not always clean or dramatic. It can happen gradually, almost invisibly, through small changes in gene frequencies within isolated populations. But in some cases, it can happen through more abrupt chromosomal changes. Errors during cell division may create individuals with unusual chromosome numbers. In plants especially, such changes can produce polyploid individuals with doubled chromosome sets. These organisms may become reproductively isolated from their parent population almost instantly. In one step, a new species can emerge, not through long geographic divergence, but through a genetic shift so large that ordinary interbreeding is no longer possible. This reminds us that evolution has more than one tempo. Sometimes it is slow and branching. Sometimes it makes sudden jumps. Random genetic drift adds yet another force, especially in small populations. Not every change in allele frequency is driven by adaptation. Sometimes chance alone is enough. In a small breeding group, An allele may become more common simply because its carriers happened to reproduce more in one generation, or less common because they happened not to. Over time, these random fluctuations can reshape a gene pool in ways that have nothing to do with advantage. This is one of the most humbling insights in population genetics. Evolution is not always a march toward better adaptation. Sometimes it is partly the result of statistical accident. Put all of these forces together: selection, mutation, gene flow, isolation, drift, and the population stops looking like a stable unit. It becomes a field of tension. Some forces hold it together, others pull it apart. Some create variation, others remove it. Some preserve similarity across space, others generate divergence. Evolution at this level is not a single mechanism, but the combined outcome of many pressures acting at once on the same gene pool. And this is where population genetics becomes so beautiful. It takes Darwin's sweeping insight that species change through time and gives it a fine internal structure. It explains not only that life evolves, but how populations carry the machinery of that change within themselves. Species are no longer just names in a book or branches on a diagram. They are living gene pools, always in motion, always susceptible to balance or disruption, always capable of splitting, adapting, or disappearing. In the end, a new species does not appear because nature decides to invent one. It appears because a population changes for long enough, deeply enough, and separately enough that interbreeding with the ancestral stock is no longer possible. That is the quiet threshold where one lineage becomes two. No trumpet announces it. No single generation sees the full transformation. But over time, the gene pool tells the truth. The frequencies shift, the barriers rise, the old continuity breaks. And from that break, biological novelty enters the world. Natural selection can feel abstract when it is explained only as an idea. Variation exists. Some individuals survive better than others. Traits spread through reproduction. On paper, the logic is clean. In the real world, it is often much harder to see because most environments do not change fast enough for the process to announce itself dramatically. But under intense pressure, evolution becomes visible. It stops feeling like theory and starts looking like an event. That is what makes the classic examples of natural selection so compelling. They do not merely suggest that adaptation happens, they show populations changing while the selective pressure is still active, sometimes within a span short enough for human observers to witness it. And when that happens, nature becomes startlingly clear. The environment does not create what it needs from nothing. It exposes variation that is already present, then filters it with ruthless consistency. One of the clearest examples comes from insecticide resistance. Insects are almost perfectly designed to reveal evolution under pressure, not because they are unusually special, but because their biology accelerates the process. They reproduce quickly, often in enormous numbers. They occupy nearly every environment imaginable. Their populations contain variation, and that variation is constantly being tested. When an insecticide such as DDT is introduced, the result at first appears overwhelming. Large numbers of insects die, disease transmission drops, agriculture damage decreases. For a moment, it can seem as if the chemical has solved the problem completely. But only for a moment. The crucial detail is that no natural population is perfectly uniform. Even before the chemical appears, individuals differ. Some are extremely susceptible. Some are moderately susceptible. A few, by chance, carry traits that make them more resistant. In an untreated world, those differences may not matter much. Under heavy insecticide exposure, they suddenly matter more than almost anything else. The susceptible die first, the resistant survive, and because the survivors now make up a larger fraction of the breeding population, the next generation contains more of the genes associated with resistance. This is directional selection in one of its purest forms. The environment is no longer neutral. It is pushing the distribution of traits in one clear direction. What matters here is not that resistance appears out of nowhere at the right time. What matters is that the environment changes which existing variants get to reproduce. A population that once contained only a small minority of resistant individuals can, after repeated exposure, become dominated by them. The chemical did not invent the resistance, it selected for it. That distinction is one of the most important in all of evolutionary biology. People often imagine adaptation as if the environment somehow instructs life to change in the needed direction, but the evidence points elsewhere. Natural selection works because variation already exists. It does not design, it filters. The history of DDT made this painfully obvious. Early results were dramatic enough to inspire near total confidence. Typhus cases fell sharply where rat fleas were controlled. Mosquito populations collapsed in sprayed regions. Agricultural pests seemed vulnerable at last. Yet by the late 1940s and 1950s, signs of resistance were already emerging. Houseflies, mosquitoes, and many other insect species began to rebound, not because the toxin had become weak, but because the populations had changed. The breeding population was no longer the same population the insecticide had first attacked. This is one of the most elegant and unsettling demonstrations of natural selection. The selective agent remains constant, but the population evolves around it. What makes this so interesting is that natural selection can be observed statistically long before it becomes visually dramatic. If the original population has a broad range of susceptibility, the first application of an insecticide wipes out the most vulnerable fraction. The survivors cluster toward the resistant end of the distribution. Repeat the pressure across several generations, and the whole curve shifts. A population that was once mostly vulnerable becomes mostly resistant. In evolutionary terms, this is a change in gene frequency made visible by environmental pressure. Yet there is something else equally fascinating here. Once the strong directional pressure is removed, natural selection does not always continue pushing change in the same way. In more stable conditions, populations may move toward what biologists call stabilizing selection. Extreme variance at either end of a trait distribution may be reduced, while intermediate forms become more common. In this sense, natural selection is not always a force of dramatic innovation. Sometimes it acts as a normalizing process, preserving a population around a more stable center. Another classic example comes from industrial melanism, and few stories in evolutionary biology are as visually striking as this one. In the peppered moth of England, two color forms existed, a light form and a dark melanic form. Before industrial pollution darkened the landscape, tree trunks in many regions were pale and covered in lichens. On such backgrounds, the light-colored moth was harder for predators to see. The darker form existed, but it was rare. As industrialization spread, soot and pollution transformed the environment. Tree trunks darkened, lichens declined, and the visual balance reversed. Now the light form stood out, while the melanic form became better concealed. This might have remained a suggestive story rather than a convincing one if it had not been tested so carefully, but the pattern was supported by both population data and field experiments. Over time, the frequency of the dark form rose sharply in polluted regions, while the lighter form remained common in cleaner, non-industrial areas. More importantly, experiments showed that bird predation could account for the shift. Moths that were more visible against their background were recaptured less often because they were more often eaten. Here again, the environment did not manufacture a new type on command. It changed the survival value of types already present. That is what makes industrial melanism so powerful as evidence. It is evolution reduced to a stark visual contest between visibility and concealment. A small inherited difference in color becomes the basis for differential survival, and over time, that differential survival changes the population itself. The story feels dramatic because it is easy to picture, but its logic is the same as in insecticide resistance. Variation exists first, pressure acts second, reproduction carries the result forward. By the time these examples are placed side by side, natural selection begins to look less like a vague background principle and more like a measurable force. It requires observable variation. It requires differential survival or reproduction, and it requires inheritance. When those three conditions are met, populations do not remain still. They shift. This is the deeper lesson hidden beneath both the moth and the insecticide. Natural selection is not merely about life surviving hardship. It is about populations being sorted by that hardship in ways that alter the future. The individual lives or dies in the present. The population changes in the next generation. And that is where the real drama begins. Because once biologists understood that natural selection could be observed in living populations, a second question became unavoidable. If resistant insects and darker moths were not being created by the environment in real time, then where did the favorable variants come from in the first place? What is the actual source of evolutionary novelty? Does the environment cause useful mutations to appear when needed, or does it merely choose among changes that arise on their own? That question would lead biology into one of its most elegant experiments and one of its clearest answers. Once the evidence from insecticide resistance and industrial melanism is understood, the next question becomes impossible to avoid. If the environment does not invent the traits it needs, then where do those traits come from? How does a population suddenly contain moths dark enough to blend into soot-blackened trees, or bacteria capable of surviving an antibiotic that has never touched them before? This is where the story becomes even more interesting, because it forces biology to separate two processes that are often confused. Mutation creates variation. Natural selection chooses among it. The order matters. For a long time, this was not obvious. One of the most intuitive alternatives came from the older Lamarckian view, the idea that organisms could acquire beneficial traits in response to need and then pass those traits on. It is easy to understand why this idea felt attractive. A giraffe stretches toward high leaves, so its neck becomes longer. A harsh environment appears, so a useful trait emerges in response. The logic feels direct, almost satisfying, because it gives the environment an active, instructive role. But nature is not that cooperative. The environment does not reward effort with inheritance. It does not build the right mutation on demand. It reveals which variants were already hidden in the population and then changes their value. This distinction became clear through work on bacteria and antibiotics, where evolution could be watched at extraordinary speed. When antibiotics are introduced to a bacterial population, the common impression is that the chemical somehow forces bacteria to become resistant. Resistance appears after exposure, so it is tempting to imagine that exposure caused the mutation itself. But that is not what the evidence shows. The resistant cells were already there, rare and almost invisible, before the antibiotic arrived. The drug did not create them. It simply erased their competition. This is what makes the classic bacterial experiments so powerful. They were designed to answer a very specific question. Do adaptive mutations occur after an environmental challenge appears, or do they arise randomly beforehand? The logic is elegant. If bacteria only mutate into resistant forms after they encounter an antibiotic, then separate populations exposed under similar conditions should produce roughly similar numbers of resistant colonies. But if mutations occur randomly before exposure, then the results should vary wildly depending on when the mutation happened in each growing population. A mutation that occurs early will give rise to many resistant descendants. A mutation that occurs late will produce only a few. The pattern of survivors, in other words, can reveal the timing of mutation. This is where the Liederberg experiment becomes so important. By taking bacterial colonies grown in one place and transferring their pattern onto plates containing streptomycin, the experiment showed that resistance did not appear everywhere exposure occurred. Instead, resistant colonies emerged in the same spatial positions where the pre-existing resistant descendants had already been growing before the antibiotic was ever introduced. The antibiotic was not generating the solution, it was uncovering it. That is a subtle difference in language, but a massive difference in biology. And once that distinction is understood, natural selection becomes even more impressive. It does not need foresight. It does not need to aim. Mutations occur randomly with respect to need. Most provide no advantage in a given environment. Some are harmful. Some are neutral. A very small number become useful when conditions change. When that change arrives, the population already contains the raw material for adaptation, scattered through individuals like hidden cards in a shuffled deck. Selection then acts with brutal simplicity. It removes the unfit under the new conditions and allows the already advantaged variants to dominate the next generation. This is why natural selection is so often invisible until the environment changes sharply. In stable conditions, a resistant bacterium may have little advantage at all. A dark moth may be no better than a pale one. A resistant insect may be only one rare form among many. But change the background, introduce the toxin, alter the pressure, and what was once marginal suddenly becomes decisive. The environment does not write the mutation, it changes the meaning of the mutation. That may be the most important lesson of all. Natural selection is not creative in the way human intuition wants it to be. It does not stand above life choosing future designs. It is reactive, statistical, and unsentimental. It operates on what already exists. Yet over time, that is more than enough. If variation is constant and reproduction is continuous, then even a blind filter can produce extraordinary adaptation. There is also a deeper consequence hidden in this mechanism. Because mutation comes first and selection follows, populations always carry more potential than is immediately visible. A trait that seems useless today may become critical tomorrow. A mutation that drifts silently through a population may turn into a survival advantage when the environment changes. Evolution, then, is not just a response to the present. It is the continual reshuffling of possibilities that may reveal their importance only later. This helps explain why natural selection can operate so quickly in some contexts and so slowly in others. In bacteria, insects, and short-lived organisms, the cycle of mutation selection and reproduction can unfold within years, even months. In larger animals, the same process may take thousands of generations, but the underlying logic does not change. Variation appears first, selection acts after. Populations shift, gene frequencies move. Adaptation spreads, not because nature planned ahead, but because enough variation already existed for selection to work with. That is also why natural selection should never be confused with perfection. It does not build ideal organisms. It favors what works well enough under current conditions. What succeeds in one environment may fail in another. DDT resistance becomes a triumph under chemical pressure, but may carry costs in a toxin-free world. A dark moth becomes advantageous in an industrial forest and vulnerable again when clearer air returns and lichens reclaim the bark. Selection is always local, always conditional, always tied to the momentary structure of survival, and that makes it even more real. Because in the natural world, there are no final winners. There are only populations passing through changing conditions, carrying variation forward, being tested again and again by environments that never stay still for long. This is what the evidence of natural selection ultimately reveals. Not merely that life changes, but how. Observable variation exists within populations. The environment does not treat all variants equally. Survival and reproduction become uneven. Over time, that unevenness alters the composition of the population. Whether the setting is a polluted forest, an insect population under chemical attack, or a bacterial colony facing antibiotics, the principle remains the same. The selective pressure does not need intelligence, it only needs consistency. And from that consistency, entire populations can be transformed. So the story of natural selection is not really about strength in the heroic sense, nor about life being rewarded for trying. It is about probabilities shifting under pressure. It is about hidden variants becoming decisive. It is about populations being edited by reality itself. The mutation appears first, quietly, without purpose. The environment changes second, without mercy. And then survival begins choosing. Every story about life begins with a deeper question. Before the first cell divided, before the first ocean carried organic molecules, before the earth had continents, clouds, or even a solid crust, there had to be a universe capable of producing such a world at all. Makes the study of origins so fascinating. It does not begin with biology. It begins with fire, gravity, chemistry, and time on a scale so vast that ordinary language struggles to contain it. From Earth, the night sky can feel calm and distant, a quiet ceiling of light. But that appearance is deceptive. Stars are not ornaments scattered across darkness. They are immense nuclear furnaces, bodies of hydrogen and helium burning through fusion, producing the energy that makes planets visible and life conceivable. They are grouped into galaxies, vast systems of matter held together by gravity, and those galaxies themselves reveal that the universe is not static. Some regions contain aging stars already nearing their end, while elsewhere, new stars are forming inside immense clouds of gas collapsing under their own weight. The universe is not a finished structure. It is a continuous process of formation and transformation. Modern cosmology places the beginning of that process roughly 13.8 billion years ago in an event where the matter and energy of the universe existed in an extraordinarily compressed and unstable state. From that beginning, matter expanded, cooled, and began to organize. Galaxies formed. Within them, clouds of gas condensed into stars. Around some of those stars, smaller residual accumulations of dust and debris gathered into planets. Our own solar system emerged from this larger story, not as a separate miracle, but as part of a chain of cosmic events already unfolding long before the Earth existed. The Earth formed from matter left over from the birth of the Sun. At first, it was not a blue planet. It was a young, violent sphere of molten material. The heavier elements sank inward toward the center, creating the core, while the lighter materials rose outward and eventually solidified into the earliest crust. In this stage, the planet would have been almost unrecognizable, unstable, intensely hot, and wrapped in a dense atmosphere unlike the one we breathe now. There were no oceans yet, only vapor, gases, and a surface still too hostile for life. That early atmosphere is one of the most important details in the story of origin. The modern air of Earth is dominated by nitrogen and oxygen, but the primitive atmosphere was very different. It likely contained hydrogen, methane, ammonia, and water vapor, along with other gases released from the still cooling planet and from volcanic activity. In time, the Earth cooled enough for water vapor to condense. Rain began to fall, and not briefly, but over immense spans of time. The surface collected water, and the first oceans formed. This was a decisive moment. Water did not create life by itself, but it provided the medium in which the chemistry of life could begin. Meanwhile, the young Earth was anything but quiet. Volcanic chains rose above the ocean surface. Sediments accumulated in shallow basins. Subsurface pressures compressed these deposits, warping and uplifting them into folded mountain systems. Erosion wore them down. New sediments gathered again. Even before life, the Earth was already entering cycles of construction and destruction that would later shape every habitat the biosphere would ever use. Continents expanded, ocean basins shifted. The planet was building its own geological machinery. But the most profound transition had not yet occurred. A lifeless Earth, no matter how geologically active, is still only a stage. The true mystery lies in how chemistry became biology. The fossil evidence for the earliest life is frustratingly sparse, but the oldest rocks on Earth have often been buried, altered, or destroyed by the planet's own restless crust. Yet the absence of direct observation did not stop scientists from asking how life might have begun. Their reasoning followed the same pattern in astronomy and geology. Infer the past from surviving evidence, reconstruct the environment, and ask what processes could plausibly operate there. If life arose from non-living matter, then there must have been a sequence of physical and chemical events leading from simple molecules to something capable of metabolism, organization, and replication. This idea became known as inorganic evolution, and it remains one of the most intriguing concepts in science. The suggestion is not that life appeared fully formed, but that early Earth contained conditions favorable for the synthesis of organic molecules from simpler inorganic compounds. In the warm, shallow seas of the early Precambrian world, minerals washed from weathered rock mixed with dissolved gases from the atmosphere. Lightning, ultraviolet radiation, and other energy sources provided the force needed to drive chemical reactions. Under those conditions, the first building blocks of living systems may have formed gradually in the waters of the young Earth. One of the greatest turning points in this line of thought came in 1953 when Stanley Miller designed an experiment to simulate aspects of the early Earth environment. By combining methane, ammonia, hydrogen, and water in a circulating system and exposing the mixture to electrical discharge, he showed that amino acids and other organic molecules could form under such conditions. The experiment did not create life. It did something more subtle and more important. It demonstrated that the chemistry required for life did not have to be imported from outside biology. It could emerge naturally from the interaction of simple compounds under the right conditions. That result reshaped the discussion. It suggested that the first steps toward life were chemically plausible. The primitive oceans may have accumulated a growing inventory of organic precursors, molecules such as amino acids, nucleic acid components, and other building blocks that later became part of proteins, DNA, RNA, and cellular systems. What mattered was not any single molecule, but the accumulation of complexity. From there, the problem becomes one of organization. Organic molecules alone are not alive. A pool of chemicals is not a cell. Life requires more than ingredients. It requires structure, boundaries, internal coordination, and the ability to use energy. That is why the transition from chemistry to biology is so extraordinary. At some point, non-living systems had to cross a threshold where they were no longer just reacting, but maintaining themselves, reproducing patterns, and becoming subject to selection. This stage of the story remains necessarily speculative in places, yet its logic is compelling. The first forms of life were likely extremely simple, probably comparative in complexity to primitive bacterial systems. They had to meet only a few essential criteria: organization, the ability to use energy, and the capacity for reproduction with heredity. These earliest living systems would have been chemically close to their environment, feeding on organic material that accumulated through abiotic processes in the oceans around them. They were not yet masters of the planet. They were fragile outcomes of a chemistry that had become just stable enough to persist. And yet, that was enough. Because once heredity enters the picture, evolution begins. The first separation of one living system from another creates the possibility of variation. Variation creates the possibility of selection. Selection creates the possibility of divergence. In that sense, the first living cell did not merely survive, it opened the door to every later form of life. What makes the origin story so gripping is that it does not move from nothing to complexity in a single leap. It moves through stages. Cosmic matter becomes stars. Residual matter becomes planets. A molten world cools into a crust, an atmosphere, and oceans. Chemistry becomes richer, more organized, more dynamic. Molecules begin to assemble into systems. Systems become capable of maintaining themselves. And somewhere in that gradual intensification, the first true boundary between non-living matter and living process was crossed. That moment may never be witnessed directly. It lies too deep in time, too far behind the surviving evidence, but its consequences are all around us. Every forest, every animal, every cell dividing inside a living body traces back to that transition. Life did not appear on a finished world. It emerged on a young, unstable, chemically restless Earth that was only beginning to become habitable. And from that first fragile threshold, everything else would follow. The first living systems were almost certainly nothing like the world we recognize now. They were simple, microscopic, and chemically dependent on the environment around them. In a sense, the earliest organisms did not dominate the planet, they borrowed from it. The primitive oceans of the early Earth likely contained a supply of organic molecules produced through abiotic processes, and those first bacterial-like forms survived by taking in what the environment already offered. They were not yet building food in the modern biological sense. they were consuming the leftovers of a chemically active world. That makes the earliest phase of life both humble and astonishing. It was humble because these first organisms were simple, probably prokaryotic in organization, lacking nuclei and the internal compartmental complexity that later cells would develop. But it was astonishing because once even that level of organization existed, evolution gained a foothold it had never possessed before. From that point on, variation, heredity, and selection could begin shaping not just molecules, but living systems. One of the most important steps in that process was organization itself. Life is unusual not merely because it consists of special chemicals, but because those chemicals are arranged into a system that maintains itself. A living cell is not just matter, it is matter under coordination. It has a boundary that separates inner chemistry from outer environment. It uses energy. It repairs, reproduces, and preserves inherited information. That is why the origin of life is not only about the synthesis of amino acids or nucleic acids, it is about the emergence of stable organization. From these earliest prokaryotic forms, evolution eventually produced a far more complex type of cell, the eukaryotic cell. This was one of the great turning points in the history of life because eukaryotes introduced a level of internal architecture that changed what cells could become. Instead of a relatively simple cellular system, the eukaryotic cell contained specialized internal compartments, each performing different functions. Membranes surrounded organelles. A nucleus enclosed the genetic material. Mitochondria handled energy conversion. Other structures managed synthesis, digestion, transport, and storage. The cell was no longer just a chemical boundary, it had become an internal city. The origin of that complexity is one of the most fascinating puzzles in biology. One explanation, emphasized in the material, is symbiosis. Symbiosis is a close biological relationship between organisms in which both may benefit or one may come to depend upon the other. In nature, such relationships are not rare. Lichens, for example, are built from an algae and a fungus living together so closely that the partnership becomes a stable biological unit. The algae provides food through photosynthesis. The fungus provides water, minerals, and protection. Two organisms remain biologically distinct, yet function as one system. The symbiotic theory of eukaryotic origin extends this logic deep into evolutionary history. According to this idea, early eukaryotic cells may have arisen when larger prokaryotic cells engulfed smaller ones, but instead of digesting them, retained them. Over time, the relationship became permanent. The smaller cells specialized, taking on functions such as respiration or photosynthesis, while the host cell provided a protective and organized environment. The modern mitochondrion and chloroplast still carry traces of this ancestry, including their own DNA and certain independent-like characteristics. If this interpretation is correct, then the complexity of the eukaryotic cell did not arise solely through gradual internal modification. It also arose through alliance. That idea is extraordinary because it means one of the greatest leaps in biological complexity may have been built not through conquest, but through cooperation. Once eukaryotic cells existed, A new adaptive possibility opened: multicellularity. A single cell, however complex, is still limited. It can do many things, but it cannot easily become a body. With multicellularity came the possibility of specialization at a higher level. Some cells could take on one function, others another. Tissues became possible, structure became scalable. The living world was no longer limited to solitary microscopic forms. But before multicellular animals and plants could transform the Earth, another revolution had to occur. Life had to learn how to make its own food. The earliest organisms were dependent on pre-existing organic material in the oceans. That was a fragile strategy because the supply was limited. As populations increased, competition for this abiotic food would have intensified. At some point, a new method of survival became decisive: the ability to capture energy directly and use it to synthesize organic molecules from inorganic substances. This was the origin of photosynthesis, one of the most important biological inventions in the history of the planet. The first photosynthetic organisms were likely simple, perhaps bacterial in nature, but their impact was immense. By using sunlight, carbon dioxide, and water to create sugars, they freed life from complete dependence on the chemical leftovers of the abiotic world. More than that, photosynthesis released oxygen as a byproduct. At first, this oxygen would have reacted with various materials in the oceans and crust, but over long spans of time, it accumulated in the atmosphere. That gradual enrichment changed the chemistry of the planet. This oxygen revolution may be one of the most consequential events in Earth history. Early life evolved in an atmosphere rich in methane and ammonia, but the increasing presence of oxygen altered the balance of those gases and transformed the environmental conditions under which life had to operate. For some organisms, oxygen would have been toxic. For others, it became opportunity. Eventually, oxygen-based respiration would become one of the most efficient energy systems in biology, and nearly all complex life now depends on it. In that sense, plants did not merely feed themselves, their ancestors remade the air. The first plants were likely not plants in the modern forest sense, but photosynthetic organisms that gradually increased the amount of biologically available food in the world. From that moment onward, food webs could expand. Life was no longer limited by the dwindling stock of primordial organic compounds. It could be generated continuously from sunlight. Animals entered the story later, and they did so by exploiting the new possibilities created by multicellularity and food abundance. According to the material, the first animals may have evolved from colonial algae or other simple eukaryotic forms that were only marginally efficient in photosynthesis and supplemented their nutrition by engulfing the cells of more efficient species. Over time, some of these lineages abandoned photosynthesis altogether, becoming fully heterotrophic. From that beginning came the first truly animal mode of life: movement, ingestion, active searching, and predation. This transition matters because it marks a fundamental shift in strategy. Plants and photosynthetic organisms capture energy directly. Animals must pursue, gather, or consume what others have already made. The rise of animals, therefore, changed ecosystems. not just by adding new organisms, but by introducing new relationships, feeding, pursuit, mobility, and increasingly complex interactions. By the late Precambrian, multicellular life had begun to flourish. Fossils from the Ediacaran Hills of Australia preserve some of the earliest known multicellular organisms, dating to just over 600 million years ago, near the close of the Precambrian era. These forms were still relatively simple compared with later animals, yet they show that life had already moved far beyond the single-celled stage. Then, at the beginning of the Cambrian period, the fossil record seems to burst with diversity. Invertebrates became abundant enough that this interval is often called the Age of Invertebrates. Jellyfish, segmented worms, shell-bearing organisms, trilobites, and many other forms appeared in large numbers. The oceans became crowded with bodies, movement, armor, and experimentation. This Cambrian expansion was not the origin of life, but it was the moment when life became visibly elaborate. And that is what makes the whole story so striking. The history of origin is not a single mystery solved in one step. It is a sequence of thresholds. First, the universe becomes capable of stars. Then, stars yield planets. Then, the Earth cools into a world with crust, atmosphere, and oceans. Chemistry becomes organized enough to sustain the first cells. Prokaryotic life appears. Eukaryotic complexity emerges. Symbiosis creates new cellular possibilities. Photosynthesis transforms the air. Multicellularity expands the architecture of bodies. Animals begin to move, consume, and diversify. Each threshold changes the rules for everything that follows. By the time the first rich animal ecosystems appear in the fossil record, the long-hidden work of origin has already done its job. The world is no longer merely habitable, it has become biological. Oceans are no longer chemical reservoirs alone. They are ecosystems. The atmosphere is no longer only a blanket of gases, it is part of life's machinery. Cells are no longer isolated experiments, they are the building blocks of bodies, colonies, tissues, and eventually worlds dense with interaction. What began as chemistry has become history, and once life crossed those early thresholds, the Earth would never be lifeless again. If evolution explains how life changes, phylogenetic divergence explains how life separates. It is the moment when one successful form does not simply improve, but divides into many descendants, each pushed toward a different way of surviving. Over deep time, this is what gives the tree of life its shape. Lineages do not march forward in a single line. They branch, split, radiate, and specialize until entire worlds are filled with organisms that share distant ancestry but live as if they were designed for completely different planets. The Paleozoic era was one of the great ages of branching. During this immense stretch of time, invertebrates flourished, vertebrates appeared, and the first major groups of fish, amphibians, and reptiles took form. It was an era not just of new species, but of new strategies. Life was experimenting with armor, jaws, lungs, limbs, eggs, and eventually and eventually the ability to leave water behind and claim the land. What makes this so fascinating is that none of these victories appeared fully formed. Each emerged from earlier structures, modified step by step until a threshold was crossed and a new branch of life began. The invertebrates came first, and by the early Paleozoic, they were already the dominant forms in the seas. Their sudden abundance in the fossil record once posed a problem for paleontologists because many major invertebrate groups appear with bodies already highly specialized. That can make the history behind them seem hidden, as if they erupted into existence without ancestors. But the more likely explanation is not that they were born fully made, but that the older stages of their history were less easily preserved. Soft-bodied organisms fossilize poorly, and the earliest members of many lineages may have left few clear traces behind. Even so, by the Cambrian and early Paleozoic, the seas were full of invertebrate innovation: arthropods, mollusks, annelids, echinoderms, coelentrates, and more, each representing a different solution to movement, feeding, defense, and reproduction. From among these marine worlds, vertebrates eventually emerged. Paleontologists have often regarded vertebrates as having evolved from invertebrate ancestors resembling the lancelet, a small fish-like marine animal with no true backbone, but with one of the most important structures in vertebrate history, the notochord. This flexible rod runs the length of the body and provides support. The lancelet also possesses a dorsal nerve cord and pharyngeal slits, features that connect it conceptually with the earliest vertebrate condition. It is not a vertebrate in the full sense, but it preserves something close to the architecture from which vertebrates likely arose. That is what makes it so interesting. Sometimes the most revealing organisms are not the most impressive ones, but the simplest survivors of an ancient time. The first two vertebrate fossils appear in rocks in the Ordovician period, and they belonged to jawless fishes. These early forms, often grouped as ostracoderms, possessed a primitive spinal column and an internal skeleton made largely of cartilage rather than true bone. Without jaws, their feeding options were limited. They lived close to the bottom, taking in organic matter or small aquatic organisms. Yet even these modest creatures mattered enormously because they established the vertebrate body plan on which so much later evolution would depend. In a sense, they were the first proof that life had begun building a new kind of animal, one with an internal framework capable of far greater future elaboration. By the Silurian and Devonian, vertebrate evolution accelerated. Jawless fish were joined by more advanced groups, including placoderms, fishes whose jaws allowed a much wider range of feeding strategies, including predation. This was a major turning point. Jaws do not merely improve feeding, they open ecological opportunity. Suddenly, vertebrates could bite, seize, cut, and dominate in ways jawless ancestors never could. The placoderms and other armored fishes became formidable animals protected by heavy plates over the front of the body. For a time, they were highly successful, yet success in evolution is never permanent. Their dominance faded, and by the end of the Paleozoic, most armored fish had disappeared. The great winners of the aquatic world would be the bony fishes. By the late Devonian, they had established the success of the vertebrate line in a much more enduring form. Their bodies were streamlined for movement through water, their skeletons strengthened by calcified tissue, and their jaws and teeth became more efficient. Compared with many earlier armored fishes, bony fishes were lighter, quicker, and more maneuverable. They were not burdened by massive protective plating. Instead, they were built for control and versatility. In evolutionary terms, this is a familiar pattern. A heavily armored pioneer may dominate briefly, but a more flexible and efficient form often outlasts it. Sharks also emerged during this broad expansion of fish life, first appearing in the Devonian and then diversifying further in the Paleozoic seas. They retained cartilaginous skeletons and remained almost entirely marine in distribution, but their streamlined bodies and predatory habits gave them remarkable staying power. While many earlier vertebrate experiments faded, sharks endured. They are often called primitive because of their cartilage rather than bone, but that label can be misleading. Primitive does not mean unsuccessful. In some cases, it means that an ancient design was already effective enough to survive enormous spans of time with relatively little need for radical change. Then came one of the most extraordinary transitions in the history of life, the movement from water onto land. This did not happen because fish suddenly chose the shore. It happened because environmental pressures made life in shallow, unstable waters increasingly risky, and because some fishes already possessed structures that could be modified for a different world. During the Devonian, extensive droughts lowered water levels and left many organisms stranded in shrinking habitats. For a fish, life out of water presents several immediate problems. The body loses the buoyant support of water. Gills collapse and cannot extract oxygen from air efficiently. The skin dries. Movement becomes clumsy because fins are not built to hold weight against gravity. To survive even briefly on land, a fish would need ways to breathe, support itself, and move. The most important candidates for this transition were the lobe-finned fishes. Among the many fishes of the Devonian, these forms possessed an unusual combination of features that favored survival on drying margins. Their lateral fins were not delicate fan-like structures alone, but fleshy lobes containing internal bones arranged in a pattern that could bear comparison with later limb structure. Some also possessed lungs or lung-like air sacs. In shallow, oxygen-poor waters, the ability to gulp air from the surface would already have been useful. That meant one of the great challenges of land life, breathing air, did not have to appear from nothing. It could evolve by modifying a structure that already offered partial advantage in water. This is what makes the lobe-finned fishes so compelling. They are not interesting because they are halfway creatures in some simplistic sense. They are interesting because they show how major transitions become possible. A structure that begins as a fish fin becomes, through alteration, the basis for a limb. An air sac that used to supplement oxygen intake becomes part of a more advanced breathing system. Evolution rarely leaps across impossible gaps. It repurposes what already exists. The transition to amphibians depended on precisely this kind of cumulative change. The lobe-finned fish line gradually accumulated traits that better suited survival in air and on land: stronger internal support in their fins, thicker skin to reduce water loss, stronger girdles and spine, changes in the skull and teeth for new feeding conditions, and an increasing ability to use atmospheric oxygen. Over time, these modifications produced the first amphibians, vertebrates capable of venturing on the land while still retaining strong ties to the water. That dependence is part of what makes amphibians such an important stage rather than a final conquest. They represent success, but incomplete success. Adult amphibians could move across land and exploit food there, yet their reproduction remained bound to moist environments. Their eggs lacked the protections needed for dry terrestrial conditions, and fertilization often still required water. In a sense, amphibians opened the door to land without yet being able to shut the old aquatic world behind them. Even so, their arrival changed everything. Their continents were no longer empty margins beyond the reach of vertebrate life. For the first time, backboned animals had begun to establish themselves on land. The branch had split. One great line of vertebrate life would remain in the waters. Another had begun the long and difficult process of becoming terrestrial. And once that threshold had been crossed, the history of divergence would accelerate again. Because the first amphibians were not the end of the story, they were only the beginning of the next world. Amphibians were the pioneers of the land, but they were not its final conquerors. They could move, hunt, and breathe in the new terrestrial world, yet they remained tethered to moist environments by reproduction. Their eggs were vulnerable to drying. Their life cycle still carried the memory of water. This meant that even after vertebrates had made the dramatic leap on the land, they had not yet become fully independent of the world they came from. The true liberation of vertebrate life on land required a second innovation, one just as decisive as limbs and lungs, a way to reproduce without returning to water. That breakthrough came with the reptiles. Emerging late in the Carboniferous and expanding through the Permian, reptiles represented the first completely terrestrial vertebrates. Their success did not rest on a single trait, but on a suite of advances that together changed the rules of life on land. Their skulls became stronger and more specialized. Their skin grew tougher, better able to resist water loss. Their limbs and girdles became more effective at bearing weight. Their nervous systems and sensory adaptations improved. But above all, the great innovation of the reptilian line was the amniot egg. The amniot egg is one of the most important inventions in the history of life. It carried its own protected aquatic environment inside a shell, allowing the embryo to develop on land without being exposed directly to desiccation. Around the embryo lay membranes performing separate functions: protection, nutrition, waste handling, and gas exchange. In one structure, evolution solved the central reproductive weakness of amphibians. Vertebrates no longer needed ponds, marshes, or wet soil to complete their life cycle. With the amniotic egg, they could colonize drier uplands and spread into habitats that had previously remained beyond reach. That single innovation transformed divergence. Reptiles expanded widely and diversified into many forms, becoming the dominant terrestrial vertebrates of much of the later Paleozoic and Mesozoic. What followed was not one reptilian destiny, but a branching radiation into multiple lineages, each exploring different ecological strategies. Some remained small, others grew immense, some returned to the water, others took to the air, and among them, one lineage would produce the most famous land animals in Earth's history: the dinosaurs. Dinosaurs were not important merely because they were large, although many certainly were. Their significance lies in how completely they came to dominate terrestrial ecosystems for an immense span of time. They were part of a broad adaptive radiation of reptilian life, and their success rested on traits that made them effective in a wide range of conditions. Some lineages evolved toward gigantic body size, becoming the largest animals ever to exist. Others were fast, predatory, agile. Their diversity itself is the point. Once the reptilian line had freed itself reproductively from water, it was able to explore terrestrial life on a scale amphibians never could. And yet, even this age of reptilian dominance was not a single straight line. Within the broader reptilian radiation, two especially important lineages emerged during the Jurassic. One led to the pterosaurs, the flying reptiles that remained fully reptilian in structure while mastering the air. The other led toward birds. Fossils such as Archaeopteryx remain so iconic because they preserve a mixture of traits that make historical sense: feathers and flight-related adaptations alongside unmistakable reptilian features such as teeth, claws, and a long bony tail. This is exactly the sort of combination one expects during divergence. New branches do not appear fully purified of their ancestry. They carry the past forward while modifying it. Birds became one of the most successful vertebrate groups, not because they could fly, but because they combined multiple advantages: the reduction and lightening of the skeleton, the evolution of feathers, high metabolic rate, powerful flight muscles, and the emergence of warm-bloodedness together opened an enormous adaptive zone. Flight gave access to new environments and escape routes. Warm-bloodedness allowed activity across wider climatic conditions. In birds, reptilian ancestry did not disappear. It was transformed into something capable of extraordinary flexibility. Mammals arose through a different branch of the reptilian stock, descending from therapsid ancestors. The earliest mammals were small and appeared while reptiles still ruled the land. For a long time, they lived in the margins of a dinosaur-dominated world. Yet hidden inside this smallness were a series of innovations that would later prove revolutionary. Mammals evolved hair, mammary glands, a tendency toward larger brains relative to body size, and warm-blooded physiology. Like birds, warm-bloodedness gave them greater independence from environmental temperature and allowed more continuous activity. But mammals also changed reproduction and parental care in ways that deeply affected behavior. In mammals, the relationship between parent and offspring became more prolonged and biologically intimate. Milk meant nourishment could continue after birth. In more advanced forms, internal development extended further inside the mother. This changed not only anatomy, but social behavior and survival strategy. Mammals were not simply reptiles with fur. They were a new adaptive experiment in metabolism, reproduction, and intelligence. Three great mammalian groups survived into more recent times: monotremes, marsupials, and placentals. Monotremes, represented today by animals such as the platypus and echidnas, remain especially fascinating because they preserve a strange combination of features. They are mammals in possessing hair and mammary glands, yet they still lay eggs like their more ancient reptilian ancestors. In them, the branching nature of evolution becomes vividly clear. New forms do not always erase old traits completely. Sometimes they retain them, producing organisms that feel almost like living mosaics of deep history. Marsupials represent a more fully mammalian condition, yet still one distinct from placental mammals. Their young are born at an early age of development and continue much of their growth in the pouch. This mode of reproduction shaped their radiation across regions such as Australia and parts of the ancient southern continents. Geographic isolation played a major role here. As continental masses separated, mammalian lineages became isolated and followed different evolutionary paths. In South America and Australia, marsupials diversified widely before placental mammals later invaded some of these regions more extensively. Placentals, in general, pushed mammalian development further. With longer internal gestation supported by the placenta, their young were born in a more advanced state, and this reproductive system supported a vast adaptive radiation. The placental mammals eventually diversified into the familiar major groups we know today: primates, carnivores, hoofed mammals, rodents, bats, whales, and many more. Their success illustrates a larger truth about phylogenetic divergence. Once a lineage gains a major adaptive advantage, it can split repeatedly into forms that occupy almost every conceivable niche. Yet vertebrate divergence is only half the story. The rise of land animals would have been impossible without plants transforming the continents first. Plants are foundational not simply because they produce food, but because they engineer environments. Their roots stabilize soil, their tissues build structure above ground, their photosynthesis releases oxygen and powers food webs. The earliest plant ancestors were algal, and algae themselves display a range of organizational levels from single-celled to colonial to filamentous forms. This alone is interesting because it shows again how divergence proceeds through experimentation in structure. Not every lineage becomes large or complex immediately. Many remain simple while others branch toward greater organization. Bryophytes, mosses, and liverworts represent one of the earliest land plant grades. They are more complex than algae, yet still limited. They lack true vascular tissue, meaning they remain tied to moist habitats and low-growth forms. Their reproductive cycle still reflects dependence on water because sperm must swim to the egg. Even here, though, terrestrial life is advancing. The plant body is beginning to adapt to air, substrate, and gravity. The Great Leap came with vascular plants. Once plants evolved tissues specialized for conducting water and dissolved minerals upward and food downward, the entire architecture of terrestrial vegetation changed. Stems could rise higher, leaves could expand, roots could penetrate more deeply. With vascular tissue came not just transport, but support. Plants could now build bodies capable of competing for light and surviving farther from constant moisture. From these early vascular lines came several important branches: lycopods, horsetails, ferns, and eventually seed plants. Ferns still reproduced through spores and retained some dependence on water for fertilization, yet they already showed a more advanced plant body with roots, stems, and leaves. Seed plants pushed independence much further. In pteridosperms, gymnosperms, and later angiosperms, the evolution of pollen and seeds allowed reproduction to become more fully terrestrial. Pollen protected and transported the male gamete without requiring free water. Seeds enclosed the embryo in a resistant structure with stored food, allowing survival through unfavorable conditions. Gymnosperms, including conifers, cycads, and ginkgos, dominated many landscapes for immense stretches of time. Their seeds were not enclosed in fruits, but they were powerful enough as reproductive structures to allow wide colonization of upland habitats. Then came the angiosperms, the flowering plants, perhaps the most advanced vascular plants in terms of reproductive sophistication. Flowers concentrated reproductive parts into specialized structures. Pollination could be carried by wind or increasingly by animals. The ovule became enclosed within tissue that later matured into fruit, protecting the seed and often aiding its dispersal. In angiosperms, plant divergence reached a new level of ecological intimacy. Reproduction became linked not just to air and water, but to insects, birds, mammals, and whole webs of interactions. This is one of the most beautiful dimensions of phylogenetic divergence. As lineages branch, they do not simply become different, they create new worlds for one another. Plants reshape the land, allowing animals to follow. Insects and flowers co-evolve. Forests create habitats for vertebrates. Seeds become food, and food becomes opportunity. Branching is not just separation, it is the multiplication of relationships. By the time life had produced fishes, amphibians, reptiles, birds, mammals, birophytes, ferns, gymnosperms, and flowering plants, the earth had become something entirely unlike its earlier selves. The tree of life was no longer a thin structure with a few experimental branches. It was vast, tangled, and rich with adaptive history. Every major group carried the marks of both inheritance and divergence. A limb could become a wing, a grasping hand, or a walking leg. A reproductive system could remain tied to water or break free through the amniotic egg or the seed. A photosynthetic lineage could stay simple in the water or rise into forests that cover continents. That is what phylogenetic divergence really is, not just branching, but branching with consequence. It is the reason life became more than a chain of replacements. It became a multitude. One lineage enters a new world, another splits away, a third inherits an old structure and makes something astonishingly new from it. And over deep time, those countless departures fill the earth with the bewildering variety we now take for granted. Life did not become diverse by moving in one direction. It became diverse by learning how to divide. Human evolution begins with a strange and humbling fact. Before humans were toolmakers, builders, and storytellers, they were primates. And before they were primates, they were mammals shaped by older forces still deeper in time. That means the human body did not appear as something separate from nature. It emerged from it, carrying forward the architecture of earlier life, while gradually modifying it into a form capable of doing something unusual. A primate hand that once gripped branches would one day shape tools. Eyes that once scanned forest canopies for movement would later read symbols and maps. A body built for climbing would slowly be rebalanced for walking upright. Human evolution, in that sense, is less a sudden rise than a long anatomical rearrangement. The earliest primates likely resembled tree shrews, small mammals living in the forests of southeastern Asia. They descended from primitive insectivorous stock and entered a world where life in the trees favored a new suite of traits. In the branches, survival depended less on smell and more on vision. Depth perception mattered. Hands and feet had to grip rather than merely push off the ground. Limbs became more flexible. The shoulder and hip joints gained freedom of movement. The thumb and great toe began to oppose the other digits, making branch grasping and fine manipulation possible. The face flattened, the snout shortened, and the eyes moved forward, producing overlapping fields of vision and better stereoscopic depth perception. Even before anything recognizably human appeared, the primate body was already drifting toward a new sensory and mechanical world. What is so fascinating about this shift is that many of the traits later associated with humanity did not begin as human at all. They began as arboreal solutions. A hand capable of manipulating objects is first a hand capable of securely holding branches. A more mobile shoulder is first useful for climbing. Forward-facing eyes are first useful for judging distance in a three-dimensional environment full of unstable footholds. Intelligence, curiosity, and fine control did not emerge in an empty abstract space. They were built on the demands of life in trees. From these early primate lines, two broad groups emerged. One was the prosimians, a more primitive branch that included forms such as lemurs and lorises. The other was the anthropods, the line that would eventually give rise to monkeys, apes, and humans. Within the anthropods, three major groups took shape: the Old World monkeys of Africa and Asia, the New World monkeys of Central and South America, and the great apes and humans. Already the primitive tree was branching, and with each branch came new patterns of anatomy, behavior, and environment. This is where the story becomes more difficult and more interesting. To reconstruct human evolution, scientists must decide what it means to be human in the first place. That may sound obvious until one tries to apply it to fragmentary fossils. A single skull fragment, a jawbone, part of a pelvis, or the angle of a femur can become the basis for deciding whether an extinct creature was more ape-like or more human-like in life. The challenge is that evolution does not respect neat categories. Fossils often carry mixtures of traits. One feature may look modern, another ancient. One species may walk more upright than an ape, yet retain a small brain and primitive teeth. The boundary between ape and human is therefore not a single moment, but a zone of transition. Darwin had proposed that one of the first major steps toward humanity was erect posture. The logic is strong. Once locomotion on the ground began to matter more, the skeleton would have to change. An arboreal primate typically has long, gently curved forelimbs and hind limbs of nearly similar function. In a ground-dwelling, upright form, the balance shifts. The lower limbs take over the work of weight-bearing and locomotion. The arms are freed from the task of support. That one change alters almost everything. The spine, pelvis, knee, foot, and skull must all adjust to the demands of bipedal posture. The difference between ape and human postcranial skeletons reflect this beautifully. In apes, the limbs are adapted for climbing and knuckle walking, with a body plan less specialized for sustained upright walking. In humans, the lower limb becomes straighter and more load-bearing. The pelvis becomes shorter and broader, creating a different center of gravity and improving balance during walking. The wheelbone of the foot grows larger and more adapted to heel strike, helping the foot function as both landing platform and lever. The human spine develops curves that help keep the trunk balanced over the hips. All of these changes indicate that the body is no longer merely exploring the ground, it is committing to it. The skull tells an equally important part of the story. Because jaws and teeth reflect both diet and behavior, and because the skull houses the brain and sensory structures, changes here are often deeply revealing. Ground dwellers generally have coarser diets than tree dwellers, and this difference shows up in the teeth and in the way the jaws become worn during life. But in the human line, the trend is not simply toward more powerful chewing. In fact, one of the most striking patterns is reduction. The overall dentition becomes less ape-like, the canines shrink. In many apes, especially males, large canines serve as functional weapons in threat and combat. In humans, that role disappears. Teeth become shorter and less specialized for fighting. As the teeth become smaller, the jaw also changes. The face flattens further, and the powerful chewing muscles associated with large jaws diminish. This is one reason the sagittal crest, a ridge that serves as an attachment point for large jaw muscles in many apes and some early hominins, is reduced or absent in humans. At the same time, the cranium expands. In apes, brain volume commonly falls in the range of roughly 400 to 500 cubic centimeters. In modern humans, it rises dramatically, averaging around 1,200 to 1,500 cubic centimeters. The skull, therefore, becomes not only less projecting in the face, but larger in the cranial vault, with a more elevated forehead. These trends, upright posture, reduced canines, a flatter face, altered pelvis and limbs, expanding brain case, form the core of what scientists look for when distinguishing early hominins from apes. But fossils complicate the picture because the earliest candidates in the human line do not show all of these traits at once. The older ape record of the Miocene includes forms such as proconsul, Dryopithecus, and Egyptopithecus, part of a broader radiation of ape-like primates that spread through Africa, Europe, and Asia. From among such ape stock, one branch would eventually lead toward the hominins. The fossil, called Ramapithecus, was once considered especially important because it appeared to show more human-like dental traits, suggesting that it may represent part of the divergence toward the human line. Whether or not that interpretation survives in later science is less important here than the broader point preserved in the material. The human line is thought to have emerged from a complex ape radiation, not from some isolated or separate origin. Then come the Australopithecines, and here the story becomes much more vivid. These were a diverse group of primates occupying eastern and southern Africa roughly 3 to 4 million years ago. Although species boundaries within the group have long been debated, the classic contrast in the material is between Australopithecus robustus and Australopithecus africanus. Both show why human evolution is so intriguing. They are not fully ape, not fully human, but compelling mixtures of both. Australopithecus robustus was a stocky, strongly built form with powerful jaws and large chewing muscles, probably adapted to a largely vegetarian diet, though perhaps supplemented with small game. Its brain remained small, around 400 to 500 cubic centimeters, and its skull retained ape-like features such as a sagittal crest and pronounced brow ridges. Yet it also appears to have possessed nearly erect posture. That combination matters. It means upright locomotion could evolve before a large brain and before a face fully modernized. Human evolution was not a single package delivered at once. It was modular. Different traits changed at different rates. Australopithecus africanus appears more graceful, somewhat more human-like in the skull, and more likely to have lived as a small game hunter in open savanna regions of eastern Africa. Its brain remained modest, yet its overall proportions and posture brought it closer to the human condition than the more heavily built, robust forms. The material even suggests that these two may have occupied ecological niches not unlike those of gorillas and native African tribesmen today, an illustration not of direct equivalence, but of how divergence can produce multiple ways of being almost human at once. This is one of the most important things to understand about human evolution. It was not a ladder with one neat rung leading to the next. It was a branching experiment. Some forms may represent sidelines that eventually went extinct. Others may lie closer to the ancestry of later Homo. The Australopithecines are compelling precisely because they show how many human-like traits could accumulate while still leaving the organism deeply tied to an earlier primate heritage. By this stage, however, one thing had clearly changed. A primate lineage had begun to live differently on the ground. It was no longer defined primarily by life in the trees. Its body had been reoriented. The hands were freer. The feet were committed to support. The face was changing. The skull was beginning to make room for something larger. Whether through posture, teeth, or pelvis, the anatomy was already hinting that this branch of primate evolution was becoming something the world had not seen before. It was not human yet in the full sense, but it was no longer simply ape. And once that threshold had been crossed, the next stage would not just reshape the skeleton, it would reshape behavior, tools, intelligence, and eventually the entire planet. The Australopithecines show that upright posture came early, but they also make something else clear. Walking on two legs did not instantly produce humanity. A creature could stand nearly erect, carry a partly human-like pelvis and lower limb, and still possess a small brain, heavy jaws, and many ape-like features. This is what makes the next stage so important. At some point, one branch of these early hominins crossed another threshold. It was no longer defined mainly by posture and dentition. It began to change in the direction of greater manual skill, more deliberate tool use, and a growing dependence on intelligence as a survival strategy. This is where the genus Homo enters the story. One of the earliest candidates is Homo habilis, discovered in East Africa and dated to roughly 2 million years ago. Its very name, handyman, reflects the most intriguing question surrounding it. Was this the first true member of our genus, or simply a more advanced Australopithecine? The debate itself is revealing, because Homo habilis sits in exactly the kind of transitional zone evolution should produce. Its brain was larger than that of earlier Australopithecines. Its teeth and posture suggested a more human direction, and it appears to have been associated with the making and use of simple stone tools. Yet many of its features remained primitive. The ambiguity is not a weakness in the story, it is part of the story. Evolution rarely draws clean lines. What matters is that with forms like Homo habilis, the human line begins to look less like an upright ape and more like an organism whose survival increasingly depends on the forelimbs, on manipulation, and on the mental flexibility needed to exploit tools. Freed from locomotion, the hands could do more than grip branches or carry the body. They could alter the environment directly, and once a lineage begins to depend on tools, natural selection no longer acts only on anatomy. It begins to act on coordination, planning, memory, and social learning. Another African hominin, the so-called Johansen man from Ethiopia, pushed this story even deeper, suggesting that members of the human line may have existed earlier than once thought, perhaps over 3 million years ago. Whether every fossil belongs precisely where early researchers first placed it matters less here than the larger picture emerging from the evidence. The divergence toward humanity had begun well before the classic later members of Homo appeared, and it proceeded through multiple forms, some probably ancestral, others likely side branches. Then came Homo erectus, the first generally accepted and unmistakably human member of the genus in the material. With Homo erectus, the body begins to carry a more decisive human stamp. Brain capacity ranged roughly from 800 to 1300 cubic centimeters, still below the average for modern Homo sapiens, yet dramatically beyond earlier hominins. The teeth and jaw remained somewhat more primitive, but the overall posture, locomotion, and behavioral implications were unmistakable. This was a being fully committed to life on the ground, fully upright, and far more advanced in tool-making and cultural habit than its predecessors. What makes Homo erectus especially compelling is its range. Fossils attributed to this form have been found across Africa and Asia, including the famous Java and Peking remains. That geographic spread suggests something profound. The human line had become mobile, adaptable, and capable of surviving in widely different environments. No longer restricted to a narrow ecological zone, Homo erectus had become a traveler. It carried the human experiment out of its earlier African cradle and into broader landscapes. This expansion likely reflects more than better walking. It implies improved technology, greater social coordination, and a mind able to respond flexibly to new conditions. In the material, Homo erectus is presented as the point where the human line had definitely become large-brained and advanced enough to make and use tools in a sustained way. It survived for an immense stretch of time, from roughly 1.5 to 2 million years ago until perhaps 200,000 to 300,000 years ago. That endurance alone is striking. Homo erectus was not a brief experiment. It was a successful, long-lived human form. And yet, like every earlier stage, it was not the end. By around 90,000 years ago, the material introduces a new form, Homo sapiens neanderthalensis, the Neanderthal. Few fossil humans have been more misunderstood. Neanderthals are often imagined as crude, brutish, and somehow less complete than ourselves, but the fossils tell a more interesting story. Their cranial capacity ranged from roughly 1,200 to 1,500 cubic centimeters, overlapping and in some cases exceeding that of modern humans. They possessed large, heavy jaws, a receding chin, and a strongly built skeleton, yet in sheer brain volume, they were anything but diminished. The material presents them as a geographic and chronological subdivision of humanity derived through divergence from an earlier common stock, likely represented by the Heidelberg type. This is important because it reminds us that human evolution did not become simple once the genus Homo appeared. It remained branching. Subpopulations spread, became geographically isolated, and adapted to different climates and conditions. The classic Neanderthal line, heavily adapted to cold glacial Europe, may have been a terminal branch, one that developed distinctive traits suited to a harsh environment and in the simplest interpretation, did not contribute directly to later modern humans. Other related lines may have existed in North Africa and the Middle East. Again, the broader lesson is divergence, not linear replacement. Then came Homo sapiens sapiens, early modern humans. The earliest finds of this fully modern form appear in the Mount Carmel region of the Middle East and in southern France, where Cro-Magnon remains reveal a completely modern condition: a large brain, a high forehead, a prominent chin, and a tall stature. From about 35,000 years ago onward, the basic physical attributes of modern humanity changed very little. That is one of the most fascinating points of all. For tens of thousands of years, human anatomy has remained essentially stable. And yet, human history did not slow down, it accelerated. This is where human evolution begins to diverge from almost every earlier stage in the history of life. For most organisms, the story of change is written primarily in bodies: fins becoming limbs, scales becoming feathers, eggs becoming seeds, jaws becoming more specialized. In the human case, however, once a certain anatomical threshold was reached, the major changes increasingly shifted out of the skeleton and into culture. The great change in the recent history of humanity has not been physical evolution, but the advance of culture and civilization. That shift cannot be overstated. A larger brain did not merely create a cleverer animal, it created a lineage able to store survival outside the body, in tools, memory, social learning, tradition, and eventually language, art, and technology. Natural selection had shaped the anatomy that made such capacities possible, but once culture became powerful, adaptation could proceed through behavior much faster than through bone. Fire, clothing, shelter, hunting strategies, symbolic communication, and later agriculture and civilization all became ways of reshaping the environment rather than merely enduring it. This makes human evolution both a continuation of earlier life and a departure from it. We remain primates, we remain mammals. Our skeleton still tells the story of our boreal ancestors: upright locomotion, reduced canines, expanded brains, and hands liberated from locomotion. But our later history increasingly unfolds in a different register. The body changes more slowly, while culture changes with astonishing speed. The human line does not stop evolving biologically, of course, but its most visible power now lies in something earlier organisms never achieved at comparable scale: cumulative cultural inheritance. Seen this way, human evolution is not a straight climb from ape to modern person. It is a long, branching history of experiments in posture, diet, brain growth, tool use, and adaptation. Some branches ended, some persisted for immense spans of time and then vanished. Some may have lived alongside one another, sharing landscapes but following different destinies. Out of that branching history emerged a form whose anatomy finally stabilized enough that another kind of evolution could take center stage. That is the deeper meaning of the human story. A primate body reshaped for grasping, seeing, and eventually walking upright became the platform for something new. The hands were freed, the face flattened, the canines diminished, the pelvis changed, the brain case expanded. But these were not isolated improvements. They were parts of a slow reorganization that made possible a being able not only to survive the world, but to interpret it, remember it, and alter it. In that sense, humanity did not emerge when one fossil finally looked modern enough. It emerged when a branch of primates became so reorganized that thought, cooperation, and culture could begin carrying as much evolutionary weight as muscle, claw, or tooth. And once that happened, the story of human evolution no longer belonged only to bones buried in rock. It began to belong to memory. There is a point in human evolution where anatomy is no longer the whole story. Upright posture, freed hands, larger brains, and altered skulls matter enormously, but they do not fully explain what made the human line different. At some stage, survival itself began to deepen less on the strength of a single body and more on the coordinated power of many bodies acting together. That is where human evolution enters a different kind of territory. It becomes social. Social behavior is not unique to humans, of course. It can be found across the animal world, from the highly organized lives of ants and bees to the looser but still meaningful group dynamics of wolves, monkeys, and apes. In many species, cooperation improves survival. It helps with defense, food gathering, reproduction, and the raising of young. But in humans, social behavior crossed another threshold. It did not remain only instinctive or situational, it became cumulative. It became cultural. Knowledge could be learned, shared, remembered, and passed on beyond the immediate limits of one generation's body. That may be one of the most important shifts in all of evolution. The roots of that shift reach back into primate life. The behavior of living primates offers important clues to the earlier stages of hominid social evolution because many of the adaptive patterns seen in monkeys and apes foreshadow human tendencies. Macaques and chimpanzees, for example, can move bipedally for short distances while using their hands to carry objects. Chimpanzees have enough visual awareness and motor control to detect predators, navigate complex environments, and manipulate their surroundings in flexible ways. Gorillas and chimpanzees also show that diet and social structure are not rigidly fixed. Gorillas, though largely vegetarian, may occasionally supplement their food with small animals. Chimpanzees are capable of more deliberate and active forms of hunting. This matters because early hominids likely entered a world where acquiring enough food on the ground demanded more than solitary foraging. If small game became part of the diet, then success would depend on stalking, coordination, sharing, and communication. A lone animal can chase. A group can plan. The hunt, in this sense, becomes more than a feeding strategy. It becomes a school for cooperation. Once food depends on group action, selection begins to favor not only physical competence, but the behavioral traits that make group action possible: awareness of others, learning, memory, signaling, anticipation, even trust. Tools fit naturally into this transition. Tool use is not absent in other animals. Chimpanzees and gorillas can manipulate branches, stones, and objects from their environment. They can use simple implements and show enough intelligence to repeat useful actions. But in early humans, tool use became something more than occasional convenience. It became culture. A shaped stone could act as a weapon, a cutter, a scraper, or a symbol of learned technique. Once a useful tool is not merely discovered, but remembered, taught, improved, and carried from one context to another, behavior leaves the realm of instinctive improvisation and enters the realm of transmitted knowledge. This is one of the most fascinating ideas in the story of human evolution. Tools did not just help humans survive, they likely fed back into human evolution itself. Individuals with more adaptable hands, finer motor control, and greater intelligence would have been better tool users. Better tool users would survive and reproduce more successfully. Over time, the hand became capable of more delicate and precise operations than the hand of any other animal. That is an extraordinary feedback loop. The brain helps shape tools. Tools reward better brains and hands. Evolution and culture begin reinforcing one another. And from here, intelligence can no longer be treated as an optional trait. In a tool-oriented culture, learning becomes essential. Skills are not reborn from nothing in every generation. They must be observed, retained, repeated, and refined. Natural selection, therefore, favors not only physical survival, but the mental capacities that support cumulative behavior. In such a world, the most successful individuals are not simply the strongest or fastest. They are the ones most capable of acquiring, remembering, and applying knowledge. This is why intelligence in human evolution cannot be separated from social life. A mind is most powerful when it can operate inside a group. One individual discovers, another imitates, a third improves, a fourth carries that improvement forward. The result is more than adaptation, it is accumulation. Human culture begins to act almost like a second inheritance system, not genetic, but learned. If the gene is the unit of biological transmissions, then the word may be seen as the unit of cultural transmission. Before language reached that level, however, there were earlier signs of development. Paleontological evidence suggests that early hominids moved through recognizable phases of behavior. Australopithecines likely lived in small groups and used simple tools while still making only primitive campsites. Homo erectus seems to have developed more advanced tools and more stable occupations of particular sites, including the use of fire. Neanderthals appear as skilled tool makers, capable hunters, and perhaps already people of symbolic awareness, as suggested by burial practices. Cro-Magnon humans, in turn, were not merely hunters, but artists and ritual beings, suggesting that abstract thought and the urge to express inner experience visually had already become part of human life. That sequence is important because it shows that culture did not appear all at once. It thickened. Camps became more stable. Tools became more specialized. Fire changed the terms of survival. Hunting became coordinated. Burials suggested memory, grief, or religious awareness. Cave paintings suggest symbolism, imagination, and the desire to communicate something beyond immediate necessity. Culture, like anatomy before it, evolved in layers. Language is one of the most difficult of these layers to reconstruct because it does not fossilize. Bones can be buried, stone tools survive, pottery, burials, and artworks leave traces. Spoken language disappears into air. Yet its importance is impossible to overstate. Communication by sound exists widely among animals, but only in humanity did speech become a fully open and flexible symbolic system. Words could stand for food, danger, water, place, memory, emotion, law, belief, and eventually things with no direct physical presence at all. Once language existed, social life changed permanently. It allowed the communication of abstract concepts, the coordination of larger groups, and the preservation of cultural knowledge in far more precise form than imitation alone could achieve. And then came civilization. For long stretches of time, humans remained hunters, their movements tied to the wandering herds that supported them. Settled life was impossible under those conditions. A hunter follows food. A farmer can stay. At some later point in history, in more than one region of the world, some human populations made that transition. They learned to work with the soil, to cultivate crops, to keep herds, and to turn temporary camps into permanent settlements. Once that happened, the logic of society changed. Food could be stored, population could increase, labor could specialize, villages could become towns, towns cities, and cities civilizations. This transition did not simply make human life larger, it made it denser. Priests, artisans, rulers, builders, and soldiers could emerge because not everyone had to gather food directly anymore. Surplus created complexity. Complexity created hierarchy, organization, conflict, writing, law, architecture, and memory on scales earlier humanity had never possessed. The rise of the great river civilizations shows that civilization was not some random burst of brilliance appearing from nowhere. It was an ecological and cultural threshold reached when agriculture, settlement, and social coordination crossed at certain level. And yet, for all its power, culture is not inheritance in the biological sense. That distinction becomes crucial. Culture is learned, not embedded in genes like eye color or bone structure. It can be transferred across populations. A person may be born into one environment and learn the religion, language, and technical skills of another. This means that cultural difference is not the same as biological divergence. That may sound obvious now, yet it remains one of the most important safeguards against the dangerous misuse of evolutionary thinking. Once culture becomes central to human success, biological traits and social achievement can no longer be carelessly collapsed into the same thing. This is where the argument becomes especially thoughtful. It warns against the old errors of social Darwinism, the attempt to misapply biological selection in order to rank cultures or races morally and intellectually. Evolutionary ideas can explain adaptation, but they do not justify myths of superiority. Human populations may differ in history, environment, and cultural development, yet culture is transferable in a way genes are not. A child born anywhere can, in principle, learn the language, techniques, and beliefs of any society. That simple fact breaks the logic of biological determinism. Seen this way, human evolution enters a very unusual place. Nature shaped the primate body. Evolution freed the hands, expanded the brain, and made social learning increasingly valuable. But the culture began to do something new. It started reshaping the conditions of selection themselves. Humans altered habitats, controlled fire, built shelters, cultivated lands, formed laws, transmitted religion, philosophy, and science, and created systems of memory outside the body. In every other branch of life, adaptation remains mostly in the organism. In humans, adaptation spills outward into the world. That may be the strangest thing about our species. We do not survive only by changing ourselves. We survive by changing the environment around us and by teaching one another how to live inside that changed world. By the time human evolution reaches this point, the story is no longer about skulls, teeth, or femurs. It is about camps, tools, speech, paintings, rituals, farms, and cities. It is about the rise of collective intelligence. It is about the fact that once the human line becomes deeply social, evolution itself began to run through memory as much as muscle. And from that moment on, to understand humanity, fossils would never be enough by themselves, because bones can tell us how a creature stood, but only culture tells us how it began to mean. Once human life became deeply social, the old rules of evolution did not disappear, but they became harder to read. In most organisms, survival pressures act more directly. Climate, food supply, predators, disease, and mating success shape the future of a lineage in ways that can often be traced through anatomy and reproduction. But in humans, culture began to intervene. It softened some pressures, intensified others, and created entirely new environments that no earlier hominin had ever experienced. This is what makes the question of humanity's evolutionary future so complicated. We are still biological organisms, still subject to mutation, selection, heredity, and differential reproduction, yet we now live inside worlds built as much by language, technology, medicine, agriculture, law, and social structure as by climate or geography. That raises an extraordinary possibility. What if the next major phase of human evolution is shaped less by wild nature and more by the human-made environment itself? This question is often explored through competing hypotheses, and what makes them compelling is not that any of them offers the final answer, but that each reveals a real tension. The first is the classical hypothesis. In this view, human populations may be moving toward greater genetic uniformity because many traits once strongly shaped by differential mortality are now increasingly buffered by culture. Medicine saves individuals who might not have survived in earlier ages. Technology softens some of the environmental pressures that once acted more harshly. Social institutions also shape reproduction in ways that remain difficult to predict. The classical hypothesis suggests that if natural selection no longer removes harmful mutations as effectively as it once did, their gradual accumulation could slowly weaken the population. In other words, civilization may protect human life so successfully that some of the old filtering pressures lose their force. That is a provocative idea because it asks whether modern human compassion and technical success might reshape the long-term pattern of heredity. Yet the picture is more complicated than a simple story of decline. Another possibility is the balance hypothesis. In this view, genetic diversity may itself be an advantage, and populations with greater variability may possess stronger adaptive resilience. Rather than imagining the future as a march toward uniform perfection, This perspective sees variation as a source of strength. Biology gives this idea serious weight. In many organisms, hybrid vigor and genetic diversity can increase resilience, flexibility, and survival when conditions begin to change. What appears imperfect from one angle may from another be quietly protective. This is one of the most important lessons in modern evolutionary thinking. Genes are not permanently good or bad in any universal sense. Their value depends on context. A trait that is neutral in one environment may become useful in another. A gene that is harmful under some conditions may still persist because it is tied to an advantage elsewhere. Sickle cell related genetics is one of the clearest examples where a damaging condition in one context can also be linked to survival value in another, especially in regions where malaria is common. None of this makes suffering benign, of course, but it does reveal something essential about evolution. It is not a moral sorting system, it is a contextual one. Nature does not reward purity, it works through trade-offs. Once that insight is accepted, the future of human evolution becomes even more uncertain because the human environment is now changing with unprecedented speed. For millions of years, the human line adapted to pressures imposed mainly by climate, predators, disease, food scarcity, and local ecological conditions. Today, many of the strongest pressures come from technological and social systems. Radiation, industrial chemicals, altered diets, modern medicine, reproductive choices, dense urban living, pollution, and global mobility all create conditions unknown to earlier humans. Even if these forces do not immediately produce visible evolutionary shifts, they still reshape the landscape in which variation either matters or fades into the background. And this is where the picture becomes especially unsettling. Humans may no longer face natural selection in the older, harsher form that shaped so much of our ancestry. Yet that does not mean evolution has stopped. It may simply be entering a different regime of selection, one in which the pressures are subtler, more diffuse, and in some cases created by civilization itself. Differential breeding patterns, social stratification, and group-level assortative mating may all influence gene frequencies. A population does not become biologically static simply because lions no longer stalk its members across open savannas. At the same time, there is a crucial distinction that cannot be overstated. Culture is learned, not inherited in the genetic sense. That means enormous differences in social achievement, technical sophistication, and symbolic life do not map neatly onto heredity. This is why any attempt to treat civilization as a simple reflection of biology is not only morally dangerous, but scientifically flawed. A child born into one society can learn the language, tools, beliefs, and techniques of another. Cultural inheritance crosses biological boundaries with extraordinary ease. That is one of the defining features of being human. We carry genes, yes, but we also carry traditions, and the second system can move far faster than the first. That fact changes the scale of adaptation. Other organisms adapt mainly by waiting for the next generation. Humans adapt partly by teaching the current generation. A dangerous winter may kill a population of animals that lacks the right fur, metabolism, or migration route. Humans may survive instead by storing food, making clothing, building shelter, using fire, or altering social organization. The body still matters, but the body is no longer the only adaptive instrument. Culture becomes an extension of biology, a second survival system layered on top of the first. This makes human evolution unique, though not separate from nature. We are still part of the same biological history that produced primates, mammals, vertebrates, and the first cells, but our lineage has added something new: adaptive information that can persist outside the genome. A technique, a law, a story, a religion, a scientific principle, a recipe, a tool design, a map, or a medical practice can be passed forward without waiting for mutation or reproduction. In that sense, civilization becomes a force that competes with, supplements, and sometimes overrides the slower pace of ordinary biological change. And yet, biology never vanishes completely. It also points toward future possibilities that are both scientific and philosophical. If genetic engineering or biochemical manipulation ever gains the power to alter human heredity directly, then humanity may face a threshold unlike any previous evolutionary transition. Natural selection would no longer be the only shaping force. Deliberate intervention could enter the process, yet such power would not answer the deepest questions. It would only sharpen them. What should be changed? Toward what end? According to what idea of an ideal human or an ideal society? These are not purely technical questions. They extend into ethics, philosophy, culture, and power. This may be the most sobering point of all. Human evolution has carried us to a stage where our technical abilities may one day influence the future of heredity itself, yet the wisdom to decide how such power should be used does not arrive automatically with that power. Science can show what is possible. It cannot, by itself, decide what is right. So the future of human evolution stands at an unusual intersection. One path emphasizes the continuing action of genes, mutation, and reproduction. Another emphasizes the buffering and reshaping force of culture, medicine, and technology. A third points toward direct intervention, where humanity may consciously alter conditions that evolution once handled blindly. All three are real possibilities, and none can be understood in isolation. What remains constant is the basic truth that humans are still evolving beings, even if the process is no longer written mainly in the same way it was for Australopithecines, Homo erectus, or Neanderthals. In earlier phases, the great visible changes lay in posture, skull shape, limbs, and brain size. In the recent human story, the most rapid changes lie in culture, social structure, and the environment we build around ourselves. The skeleton has slowed, civilization has accelerated. And perhaps that is the deeper meaning of this next stage in human evolution. The species that once survived by learning to cooperate has now built societies so complex that they have become part of the selection process. We no longer live only under nature. We live under the consequences of our own success. That is why the future of humanity cannot be read from fossils alone, or even from genes alone. It will be written in the unstable meeting point between biology and culture, between inherited bodies and invented worlds, between what evolution has made of us and what we decide to make of ourselves.